Arlıyor gibi ama artık yapacak. Şöyle mi yapsam? Şöyle herhalde daha iyi. Şöyle. Hülya Hocam hoş geldiniz. Yoğunluktan konuşamadık dün hocama. İbrahim Hocam, İbrahim Çelik Hocam. Ertan Hocam da görüntü geldi. Tamam, onda kamerada sıkıntı yok. Eğer hocanız da buradaysa, bağlanırsa çok iyi olur. Merhabalar hocalarım. Çok heyecanlıyız. Çünkü gerçekten birbirinden değerli uluslararası nitelikte hocalarımızı ağırlıyoruz. Şimdiden bir eksiğimiz, hatamız olursa sayın hocalarım affola. İbrahim Hocam çok teşekkür ederiz. Sağ olun. Ertan Hocam isterseniz Manuela Hoca'nın zaten sunumu video ile olacağı için hani isterseniz biz başlayabiliriz. O zaten sunum içinde soru gelirse ben cevaplarım diye söylemişti. Siz nasıl uygun görürsünüz hocam?
Olur Erhan Hocam. Yani öbür hocalara zaten beklemeye gerek yok diye düşünüyorum değil mi? Yani onların 40 dakika 50 dakika
hocam,
sonra
şu an Amerika'daki yani 2 saat sonra sunacak hoca da şu an sabah 5. Yani evet. Onların şu an katılması pek mümkün gözükmüyor. Çünkü programı da onların ülke saatlerine göre ayarlamaya çalıştık. Ellen Hoca'nın biraz önce hesapladık. Şu an itibariyle orada sabahın 6'sı olması lazım.
Doğrudur.
Yani eğer saatler 1 saat geri alınmadıysa.
Doğrudur.
Alındıysa
evet,
sabah 5 şu an orada.
Tamam hocam.
Kaan Hocam başlayabilir miyim? Sizin için bir sıkıntı var mı?
Her şey kontrol altında Eren Hocam. Buyurun. Düzenleme Kurulu Başkanımız Doktor Eren Özden.
Thank you, Dr. Hürkan. Good afternoon, everyone, and good morning. Welcome to our dear scientist guests who are joining us from the United States, Germany, and United Kingdom. I hope you are all having a good day so far. My name is Dr. Eren Özden. I am the chair of the Congress organizing team. Today we have very important guest, leading scientists, who will be sharing some information on their valuable work. We are sure that their insightful presentations will expand our understanding regarding seeds and seed physiology. Before we get started, I would like to give a little bit of background on myself and on our work. I did my PhD studies on the effects of alternating temperatures on eggplant seeds germination physiology under Professor Ibrahim Demir's advisory at Ankara University. Then I started working here at Iğdır University as an assistant professor, and I have been here for about 3 years now. Currently, we are running 3 projects supported by the Scientific and Technological Research Council of Turkey. One of them is on examining seed maturation physiology using post-ripening applications in mature cucumber seeds. The second project is on hormonal and enzymatic monitoring of cellular degradation levels in cucumber seeds in relation to artificial aging periods. In our third study, we are working on the morphological, physiological, and molecular characterizations of wild asparagus grown naturally in the Iğdır region and the determination of the biochemical contents of asparagus shots. As a team, we will soon be working on examining the effects of post-aging alternative repair mechanisms on cell distribution using physiological, molecular, and transmission electron microscopy. So we have an exciting afternoon coming up with wonderful speakers. We would like to welcome all invited speakers and thank them all for their great contributions and accepting our invitation. I leave the word to you, Mr. Chairman, Professor Yıldırım. Thank you for listening to me. I am very happy to meet you.
Ertan Hocam, mikrofonunuz kapalı.
Sorry, yes. Good afternoon, all of you. Welcome to this session of Turkey's 7th Seed Congress. I am Ertan Yıldırım. I work in Atatürk University, Turkey. I mainly work on stress physiology of vegetable crops, and I will be chairing this session, which will run from 3 until 7:30. There will be 6 talks, and every speaker will have 40 minutes for their presentations, followed by 5 minutes for questions and answers. First of all, I would like to thank Dr. Eren Özden and Organization Committee for this perfect organization. And I would like to thank also distinguished invited speakers for attending the Turkey 7th Seed Congress. This session on different fields of seed industry will bring together presentations on seed quality, seed treatments, physiology, breeding, and gene bugs as well. Yesterday and today up to now, we have heard and learned a lot about seed sector. We are very happy and excited to listen 6 valuable and distinguished scientists here. I would like to give some information about speakers shortly before each presentation. Before that, Türkçe söyleyeyim. Burada yabancı hocalarımızın yanında gerçekten dünya çapında çok önemli Türk hocalarımız da var. İbrahim Demir hocam, Levent Arın hocam ve şu an kendisi de yurt dışında olan Nebat Sarı hocam. Onlar da şeref verdiler sempozyuma. Teşekkür ederiz hepsine. İsmini göremediğimiz varsa kusura bakmasın onlar da. Tekrar herkese hoş geldiniz diyorum. Yes, the first speaker is Professor Dr. Manuela Nagel. Dr. Nagel is professor at Leibniz Institute of Plant Genetics and Crop Plant Research. She has obtained her doctoral degree at University of Göttingen. Her researches are addressed on long-term preservation of plant genetic resources, focusing on potato, mint, barley, wheat, oilseed rape. She has also concentrated on plant stress physiology, including free radicals, antioxidants, and redox signaling. Yes, Dr. Nagel, please. Eren hocam.
Very much for this kind introduction. Thank you also very much for inviting me, and it's a great honor to talk here at the Turkish Seed Congress and about the future of gene banks. I'm Manuela Nagel from the Leibniz Institute for Plant Genetics and Crop Plant Research. We are located here in the middle of Germany. And I'm responsible or the leader of the research group Cryo and Stress Biology, and we work on conservation methods and deterioration mechanisms of seed, pollen, and clonally propagated plant. Here in this area we have this very nice campus, and in the middle of this campus there are the seed storage facilities, we have the documentation group, we have the cryo facilities, and also a huge herbarium. And I would like to go more in detail about these gene banks and would like to structure my talk as follows. At first, I would like to talk about why and how do we conserve seeds. Second, what are the specific challenges and priorities for gene banks? And third, I missed one topic, how to deal with these challenges. So come to the first topic, why and how do we conserve seeds? So, and with this, I like to go back in history several thousand years ago, and we come to the Fertile Crescent. I would like to exemplify this on barley and realize that you are here right in the middle of the primary gene pool where barley has been domesticated. This is already about 9,000 years ago where this happened. So, and barley was at this time then distributed over time. There centers of secondary domestication in North Africa, in Ethiopia, and also in Asia, which then moved back and forward, so that here over time, also to America, a huge diversity established, and barley adapted to all these different areas and regions and the specific needs. And you nowadays have actually a huge diversity of barley present. So you have the diversity in color, in shape, in awns, in the spike size, for example. Well, in general, you know even better than me the 2-row and 6-row types. All this diversity you have available and was used over hundreds of years until industrialization started. So in the middle of 19th century, the first more developed cultivars appeared and started to replace the old land races because they just had more yield or had a better stress resistant. However, so they started to replace this huge diversity of land races available, and several researchers realized this problem, and among those researchers was the Russian geneticist Nikolai Ivanovich Vavilov. So he started then to go for collection missions actually all around the world, and started to collect material, and he put it in the first gene bank in St. Petersburg. So that was at the beginning of the 20th century. Later on, other gene banks followed, so the IPK was established in 1943. Also, gene banks in the USDA followed at this time. And then in the 70s, a huge amount of gene banks were established because it was realized all around the world that our genetic resources start to disappear. And we have more than 1,700 gene banks around the world. However, that are data from 2010, so it's probably that there are more, or some are maybe also joined. There are international centers available, which you can see here. For example, the CIMMYT in Mexico, the CIP, the International Potato Center in Peru. You have the ICARA here, still located in Syria. Nowadays it's in Lebanon. You have ICRISAT in India. And you have, for example, also the International Rice Research Center in Indonesia. And then among that, you have quite huge facilities also available. The USDA has quite big gene bank. Russia is still a big gene bank. India has a own national gene bank, China, Japan, and you have the security backup here right in the north, the Svalbard Global Seed Vault. I will give you a bit more information about that a bit later on. Yeah, and among that, of course, also the IPK. So we are among one of the biggest gene banks in the world. To give that a bit, give you a bit numbers. So also from 2010. So we have 500,000 accession in the USA, around this in China. They still do a lot of collecting missions, so it's probably that they have increased their numbers already. There's about 360,000 in India, 300,000 in Russia, and as already mentioned, we are among the 10 biggest one with 150,000. There are these more specialized gene banks like the one here at IRI in Indonesia, which have just 11 genesis and 23 genotypes, so they focus really on rice. The same as, for example, here the CIMMYT in Mexico, they focus on maize and triticale, so wheat especially. However, here in Gattersleben, for example, we have a collection of more than 3,000 different species, which has its own challenges, I have to say. So here in Turkey, I became also aware you have a quite big gene bank actually. So you have gene bank since 1964. It started with the Aegean Agricultural Research Institute, where it's called nowadays like that, in Izmir. And you have this there are more than 60,000 accessions, and you have this Turkish gene bank in Ankara with more than 60,000 accessions and drying facility, storage facility, or equipment which is necessary for a gene bank. At IPK, we had also, as mentioned, the 150,000 accessions, and here you can see actually the gene bank, the seed bank manager. This is Andreas Börner. Some of you may know him, and he is also responsible for this big garlic collection of more than 23,000 accessions and the seed storage, as mentioned. Overall, we have 140,000 samples stored as seeds. So that means we do not have all the material stored as seeds. So some part of the material is still stored at the field gene bank because it doesn't produce seeds, or it cannot be maintained via seeds. This is the case for allium, for example, for the different diversity of garlic and shallot. Also for potatoes. However, potatoes are, for example, majorly maintained in slow growth storage under in vitro conditions. Those we have in our external collection in Groß-Lüswitz. And we have here in Gattersleben a smaller collection of in vitro storage of mint, of Dioscorea, for example, and also some allium plants. We majorly focus here on cryopreservation to give these plants the chance to safely duplicate it in cryo. So, and currently there are 2,300 accessions, majorly potatoes, in cryo. So, in addition, just to give you an idea what is additionally done here at IPK, of course, the seed material needs to be maintained. So we have every year a field multiplication of about 5,000 accessions. We kept all the processes, the information about the protocols within the quality management system. We are certified via this ISO 9001-2015. And to compare our material, so to be sure that really this material, what we multiplied, is the same as we collected years before, we keep this huge collection of voucher specimens. So we have more than 450,000 voucher specimens in our storage in the herbarium. And in addition, we have also a collection of seeds which is not kept viable, which is just for comparison, and a collection of spikes available that are 167,000 so-called reference samples we have here in our storage facilities. Yeah, and addition, so we follow the so-called ABS system. So the international standards for gene banks, especially for seed storage, try to recommend the ABS system. So that means we maintain an active sample, a base sample, and a security sample. The active sample is used for distribution. So that's the one which you saw in glasses. In our case, it's kept at -18 degrees, also low moisture content. The base sample is vacuumized in aluminium foil bags, also kept at -18 degrees. And the security sample actually goes to the Svalbard Global Seed Vault. And currently, we have more than 60,000 accessioned at the Svalbard Global Seed Vault. We just sent new, fresh, multiplied material to the seed vault, and therefore it's increasing every year. And it's maintained there in their 3 storage chambers. At the moment, they started to open the second chambers because the first one is filled, and will hopefully stay there for a long time until the material deteriorates and we need to replace this material. So now we come to the specific challenges and priorities of a gene bank. So according to Young B, and I completely agree with his priorities, the first priority of a gene bank, of course, should be the germplasm maintenance and reproduction. If you really need to focus because of a lack of money, this is the first what has to be done. The germplasm which is in the gene bank needs to be properly maintained and reproduced. And this is in addition or in combination also with data management. I'm aware that a lot of gene banks around the world have problems already to maintain the material, then data management already starts to be a problem. However, to document the passport data and the processes around this material is of highest importance for a gene bank. The second priority is then, of course, to make this material available for users, and so distribution is very relevant, and of course also acquisition, because most gene banks have the mandate to maintain also all national varieties which are not on the market anymore. So that's one main source for gene bank, the germplasm acquisition. However, also still collecting missions are important because we still have material around the world which are available in their centers of origin, in their centers of domestication, which needs to be collected. But here it's challenging to get the official permits to do so because we have the Convention on Biological Diversity, the Nagoya Protocol, and different regulatories from the countries which make the acquisition very hard, but it's still not impossible and very relevant to do so. So then there is the characterization and evaluation. And of course, this is a point which is very important for breeders, because if they like to choose material, they need to know which type of material they want that is available. So the data about that is very important, and of course also supportive research and the germplasm enhancement. And as I cannot focus on all the things around the gene bank and around the challenges, I would like to draw your attention to some aspects around the germplasm maintenance and reproduction, and the germplasm characterization, evaluation, and some supportive research according to that. So I come now to the germplasm maintenance and reproduction, the challenges which we have specifically here. And you have to keep in mind, a gene bank collection is extremely diverse collection, and this is connected to a very specific management where you need trained staff. And this is the heart actually of a gene bank, people who are really aware which valuable material they have and which really want to multiply and maintain this material. And so in a gene bank like we are very happy to have the different curators who care for the different collections. And here you can see the lettuce collection, for example, which has to be grown, and then the challenges is to produce seeds. They are sometimes annual or biannual. You need also different equipment and different greenhouses for this kind of multiplication. So this is very challenging. If you have another crop like here, it's wheat. Of course, it has completely different growth dynamic, which you need to follow. So different curators are responsible for that. You can see here the harvest in the field. Here there is the harvest of some Vicia species, some legumes, which is again very different. Also the harvest procedure. You need to make sure that your material is clean. So a lot of gardeners has to clean the material over the year, that there is not a mixture also of the material, that you make sure that they are as less stress as possible during the vegetation period, so that you have finally a very high seed quality and a very high germination. Also, you need to know the system of multiplication, if it's a cross-pollinator or a self-pollinator. And of course, cross-pollinator have their own challenges, because you need to apply bee or flies or wrap them here in some foils to make sure that not other bee pollinate, for example, these sunflowers. And also, you have special systems for cereals. So if you have a diverse collection, it's very challenging to maintain and to multiply all material, and what you really need is trained staff. And this diverse collection also brings you to some other challenges, and this is related to seed longevity. So as soon as you have multiplied, you have harvested the material, you have cleaned it, you made sure that you have really high quality seeds which are dried down, then you have the storage. And as I mentioned, we have the so-called ABS system, so active sample, base sample, security sample. And then we have, if there are still some seeds left, we have ambient seed storage, where we put the remnant seeds into to give them away to people who are interested for that in the first year after harvest. And with this material, we could do also some research, and that we did here. Actually, I did long time ago. So we investigated how long seeds can be stored under ambient conditions. And that was very interesting to see, because you have here, for example, brassicaceae seed, and you can see in the first years of reproduction, of course, they should have high quality, but then within few years, here in 15 years, the material dropped down, and you have here different accessions, which have also diversity itself. You can try to compare these different species by the so-called half viability period, and therefore I put these red lines here into. And a half viability period, or P50, is the time until germination drops to 50%. So you have here the 50% and here the time. So for brassicaceae, it's about 7 years. So for wheat, it's the same. It's very similar. You have also a time of about 7 years. But if you look into barley, I mean, we have focused on barley in between, it's a bit higher. It's about 8 to 9 years. And for pea, it looks like pea is less diverse in their germination, and it can last much more longer in ambient storage. So you have here 15 years. However, of course, this is not optimum gene bank storage. So I would like to draw your attention also on this optimum condition, because when we put that at lower temperature, but very important is the lower moisture content, to continuously have a low moisture content, and the international rules say something between 15 and 35% relative humidity. Then you have here our barley collection, for example, which has been tested for with 12,500 tests, and you can see that still at the beginning there's quite a broad variation in the initial germination. This is considered to be a dormancy effect, so barley produces also dormant seeds. So it's very challenging to find out the very initial quality, so you have to apply some dormancy breaking methods. And then over the years, you have again also the diverse effect of germination of this collection. And the international rules say that you need to reproduce the material as soon as the initial germination is at 85% of the initial germination is reached. And of course, this can be quite quickly, so we have to deal with these challenges, and we have to also test it quite frequently. And it would be very nice to have also more faster viability tests available for in the future, especially for bigger gene banks like us. So, and just to complete this value we have here in barley, of course, under this optimum condition, a half viability of 155 years, whereas in brassicaceae it's 85 years, for wheat it's 150 years, and for pea 227 years. Of course, that are just estimated values, and it would be very great to know if the longevity can really kept for this long time under those conditions. So another challenge, so we had already the different challenges of diversity, of the different storage approaches, of the different species in the gene bank storage, and then we have another challenge that the years differ. So also we can feel climate change here in our area. But I tried to compare different results from different years, and I found, for example, for barley, it was that in 96, when the material was multiplied in 1976, we had a half viability period of 115 years. When the material was multiplied in 1975, and you may see it, it's not very visible, but however, on the P50 you can see it, the average P50 was then just 70 years. And when we multiplied the material, and here you really can see it, in 1974, then the P50 was just 50 years. So it really depends on the environment. So in every year, there are some species which produce very high seed quality, and there are some species which doesn't like this condition at all and produce a very low seed quality. And we have to deal with these challenges and try to produce the highest seed quality we can. So however, in between all this multiplication and storage methods, of course, for breeders, it would be more important to have this characterization and evaluation data available. And this is a big problem because characterization and evaluation is time consuming. And I don't have this data from our gene bank, but it's available from the IRI, from the International Rice Research Gene Bank, and they have actually, I hope you really can see that, half of this collection are land races actually, and the other half of this collection are improved varieties, and they showed data how they characterize their material. And the blue one is 89% of this material has been already characterized. So there are specific descriptors like flowering times, like plant height available. And according to this descriptor, the whole collection has been characterized, what is extremely valuable. However, when it comes to traits where breeders might be interested in, for example, like grain quality evaluation, then you can see most part is red. So 66% of the collection has not been characterized. And only, for example, for drought and cold stress or something like that, only 3% has been characterized. And for other abiotic stresses like diseases and insects, only 12%. And this is probably very similar here at IPK. So to do the characterization and evaluation is very time consuming, and it needs specific projects. So this is a challenge definitely for the future. However, how can we deal now with these challenges? And I would like to come back to the germplasm maintenance and reproduction again. And I must tell you concerning the problem with the seed viability testing, there are already some solutions out. So for example, the International Rice Research Center, they started to initiate or to establish a LemnaTech system where they do high throughput phenotyping or high throughput germination of their whole collection, and very efficient. However, the problem is for us, for example, I'm not so sure if that would be that efficient, because we have a collection of more than 3,000 species, whereas the IRI has a collection of 24 species. So they have more homogeneous genotypes. And for us, the challenge is to really adapt this to the specific needs of the species. However, I think that may come in future. There are also other possibilities out. And in the last years, we also tried to look into this non-invasive viability monitoring, if this makes sense, if this is useful for us. To give you an overview, there are, for example, different systems available. There's machine vision system. Here you usually have the spectrum of visible light. You have a camera system, and then you get data like you can do the seed grading, you get data like color, shape, area, and usually this is analyzed via machine learning system and is well established already in seed industry. Other systems are spectroscopical methods, so you have the near infrared or the mid-infrared system, or the Fourier transmission infrared system, or the Raman spectroscopy. And for near infrared and mid infrared, it's usually you have a source, here a source available. This emits the certain in a certain wavelength, and the sample pushes back a certain wavelength which is collected. The information are collected by a detector and analyze them, and you can get quite a lot of quality measurements around that. For example, NERS is quite well established for protein content, for water content, for example, and also other composition major composition components they try to cover. Similar also Raman, you have here also laser and detector, which gives a specific wavelength, and you can try to get data about the specific seed quality. Other methods are, for example, hyperspectral reflectance. Hyperspectral means that you have very broad range of wavelengths available, which you analyze via DeepL, so via machine learning approaches. And at the end, if you look into the papers, you get correlation coefficient for single genotypes or several genotypes of 98%, so predictiveness for germination. So 88 to 100%, can this approaches predict germination, or 84 to 100%? However, as I mentioned, this is usually single genotypes and it's very hard to predict the germination for a whole collection, but I'm quite sure that there will be improvements in future. What we tried here at IPK as well is, for example, also to analyze the metabolite diversity and come to a conclusion based off these results. So I analyzed here a mapping population of 184 genotypes and did a metabolite analysis, and the highest correlation which we found is for butanoic acid. There you have 60%, so the higher the germination, the lower the butanoic acid content. It's very similar for gluconic acid, and for glycerol, that's over several species, a strong indicator for a lower germination. So the higher the glycerol content, the lower the germination, which indicates the lipid deterioration. Yeah, and adenine we found as well. However, you still can see the germination correlations are not very impressive. So for the future, I believe that a combination of this method is required to come really to a predictive germination results and to a high throughput germination test in GenBank. So another priority, another challenge which I mentioned before, is the characterization and evaluation. And I think we made a huge step forward in the last year. Here at IPK, the sequencing and genotyping approaches were massive step forward, and this is headed here by Martin Mascher and Nils Stein at IPK, and they started to genotype the whole barley collection, all the 22,000 accessions which we had in our storage. And for example, you can see here it was genotyped by GBS, so genotyping by sequencing, and you can see the via this PCA where the material comes from. So for example, from China, from India. When you look here into this map, most of the material, of course, came from Asia and Northern Europe, but you find also material from Ethiopia, and here also from the fertile questions here in green, also from Turkey and this area. So that really shows you the diversity of this material. And this is only exemplified for barley at the moment. Wheat is genotyped, the rye collections, the bean collections. So we continue with the different collections to characterize and genotype them by this approach. And the value of this approach is at the end that it supports duplicate finding. So we can focus on specific material and develop only specific material. It helps to identify unique genotypes and genes which are relevant then for breeders later on, and that we are able to do a kind of compilation of subsets for further evaluation. This is of high importance then later on for really the usability of this material. And another step forward is that we nowadays can also make sure that this data are very available. We try to follow the FAIR principle, so findable, accessible, interoperable, reusable. And these, for example, the barley data are available via the Bridge portal, and you can choose around from this 22,000 accessions, can get this genotyping data and also few phenotypic data which have been made available. And another huge step forward is that then we can go to the really precision plant phenotyping. And here at IPK, but also in other research institutes nowadays, LamnaTech technology, LamnaTech phenotyping is available. And nowadays we started also with the so-called plant cultivation hall. which gives you a new idea of precision phenotyping. And I'd like to show you a short video which gives you an idea what the plant cultivation hall might be able to do.
A highly innovative plant cultivation facility is currently under construction. The Plant Cultivation Hall. It is specifically designed for high throughput phenotyping, with plants growing under conditions that are typical to the field, but which can be imposed in a highly reproducible manner. Unlike in field trials, environmental conditions such as light input, air temperature, CO2 concentration, and air humidity, as well as water and nutrient supplies, can be precisely set and regulated. The Plant Cultivation Hall will comprise 2 different phenotyping systems in an area of almost 500 square meters. The Rhizotron system is being developed and implemented in collaboration with the Forschungszentrum Jülich. It will support the simultaneous cultivation of up to 360 plants and will enable very detailed studies of the root architecture. The other area of the hall houses the container system for the phenotyping of stands of plants of vegetation. Various camera and sensor systems are mounted on a robotic crane that can be precisely positioned. These plant imaging systems use different wavelengths and scan them in 3 dimensions. The soil in the containers resembles the natural soil structure in the field. Its temperature can be precisely controlled at different depths. The temperature and soil moisture are monitored by numerous sensors. Built-in imaging tubes.
So this facility is very great to analyze our subsets, to analyze our core set, and do really precision phenotyping. And then finally, I would come to our research on seed longevity for the future. And here, I'm very happy that I'm a part of a team of 7 gene banks actually. So together with the Norgene, who manages this project, the Embrapa in Brazil, ICRISAT in India, Inya from Portugal. So we are a team who really are interested about the longevity of material at the Svalbard Global Seed Vault. And what we try to do is to bring all the aspects of seed longevity together to analyze diversity, and therefore we have 13 crops included in this experiment, among also pea and chickpea, maize and other cereals like rice and maize and barley. And not only the species diversity, also the environmental diversity. So we will use 3 environments of reproduction, and the genotype diversity. So we will use 5 genotypes per species to come to a conclusion. And of course, this material is distributed via SMTA, including all passport data, including all information of multiplication, then stored at IPK. And finally, we will do frequent germination tests over 100 years of storage. So this is really an experiment which is done for the future, gene banks to get more information about this material available, what we have in gene banks. And with this, I like to also conclude my talk now. So I hope I covered now some aspects about gene banks. So why are we doing that? What are we doing exactly in gene banks? I'm aware that a lot of gene banks are heavily underfunded and have to deal with the first priority, the maintenance and the reproduction of the material. But I hope that most gene banks can have in future trained staff and the technology, the equipment available to maintain a huge and genotyped unique collection, which is then securely long-term stored, which is evaluated and characterized, and which is used for germplasm improvement and to finally ensure the food quality for future generations. And with this, I like to thank, of course, my co-workers here at APK, especially Andreas Börner. Another one is my colleague from the University of Innsbruck, which is especially Ilse Krane. Some data came from the so-called EU EcoSeed project. And thank you very much for your attention, and I'm open for further questions. I have added also some references, and I would like to draw your attention finally to the Seed Symposium, to the ISTA Seed Symposium. I'm responsible for the organization of this ISTA Seed Symposium, and this year next year it's supposed to be in Christchurch, but unfortunately due to restriction we had to postpone it. This will be in 2025, but we try to organize a Seed Symposium in the next year. And so far, try to keep updated if you are interested in this. This probably, so we are still in the planning phase. We want to have the meeting in November 2022. At the moment, we are looking for the location in Greece. It will be probably a symposium of 2.5 days. And we want to integrate also Young at ISTA, who will organize a half-day symposium and a conference tour. So please stay tuned, try to keep updated. There will be the ISTA Seed Symposium next year, but not in New Zealand, in another European country, and that will be most likely Greece. And so for this,
thank you, Dr. Manuela. Thank you very much for this excellent and informative presentation. I can see 4 questions for you. The first question from the Dr. Ibrahim Demir. Dear Dr. Nagel, good to see you in Turkish Seed Congress. Is there any creation that you use whether seeds should be stored in Cairo or cold storage? Yes, please, Doctor.
Thank you very much for this question. So it's also very nice to see you here in this conference. And well, there are different criteria. I would say the most orthodox seeds are commonly stored under -18 degrees according to the international standards. You probably know. However, we are still on the way to investigate the seed storage conditions. And if you may have followed the research from Hugh Pritchard, Danny Ballesteros, you can see that the fatty acids, the oil content and composition plays a big role. And so -18 might not be always optimal. So, and therefore we're thinking also about cryo storage. However, cryo storage has not been established so far very well. So we do not really know if it's the best or not. So we are on the way of investigating, and until so far it's -18 the way to go.
Thank you. And the second question from Dr. Nazim Açıkgöz. Are there any prerequisites for seed demands in gene banks? Is there any problem in international material transfer?
Yeah, thank you for the question. International rules are very complicated, so it depends on the different countries, of course, and if the country has signed the International Treaty for Plant Genetic Resources and Food and Agriculture, then the gene banks usually can deliver material via a standard material transfer agreement, and you have to sign that. And for example, Germany, we send material out and you can receive that and also then do your research on that. But there are limitations then in the utilization. I think you cannot use it for pharmaceutical development. That requires then additional rules, but this is a very specific topic and it depends a bit on what you want to do with the material at the end. But if you want to use it just for research, it's usually open via this SMTA. I hope this answers your question.
Okay, thank you very much. And the third question from Dr. Levent Arın. Hello, Dr. Nagel. I visited your institution long time ago as scholarship student. Can you explain a little bit about how recalcitrant seeds are stored and multiplied?
Yeah, so we do not have recalcitrant seed here. However, we have material here what either does not produce seed or which cannot be maintained via seed. This is the potato, and the allium, the garlic, does not produce seed. So recalcitrant seed is rather similar. So this seed is not storable because it cannot be dried down and stored at -18 degrees. And all of this material usually have to be maintained as a living plant. And it can then go into different storage conditions. So you can have as the field multiplication. So like it is often the case for mango, for example, you have mango trees out, or for coconut trees that are recalcitrant seeds. Then the collections who keep this field gene banks try to back them up in vitro, so as very small plantlets. However, the methods need to be also developed. That's quite complicated. And I know from a recent paper from the gene bank in Leuven, they developed a protocol for coconut in vitro storage, and that was a huge breakthrough. And then, of course, we try to back up, so all of the gene banks who have this clonal collection try to back up that on cryo.
Okay, thank you very much. Last question from Ünal. Thank you, Manuela, for your nice presentation. I just have two questions. How many gene banks are there in your country, and how many genetic materials are conserved in these gene banks?
Yeah, it's actually a good question. I don't really know exactly. So we are in Gartesleben the only one for agricultural plant. However, there's also one for grape, for example, for fruit collection, so that they keep the apple collection, for example. I would estimate that Germany, with botanical gardens together, something like, so it's really estimates 300,000 accessions. So we have 150,000. And I would assume that plant genetic resources may be more, so like something like 300,000, but I never really checked that. I hope it's okay.
Thanks. Thank you very much again for your presentation. Is there any question? I think no. Thanks again. Thank you very much, Dr. Manuela.
Thank you very much.
It's very nice to see you hear that. Yes. I think we have started a little late and changed the place of questions and answers, so program has changed. We are very sorry for that. Apologies for this inconvenience. The next speaker, Professor Gerard Lebner. I can't see him in the list. Burada mı acaba Doktor Gerard Lebner?
Evet hocam. Evet yes. Hocam. Burada mikrofonu da açık.
Öyle, okay.
I'm moving.
Okay. Welcome.
Can you hear me?
Welcome. Yes, we can hear. First, I'll introduce to you a little bit, so I can give the word. Dr. Lebner.
Can you hear me?
Yes.
Okay.
Yes. Dr. Lebner is a professor and chair of plant biochemistry. He is at Royal Holloway, University of London. He has obtained his doctoral degree at Ruhr University, Bochum, Germany, and Swiss Federal Institute of Technology, Zurich. His researches are addressed on germination physiology and dormancy of plants. He has contributed his relevant fields with over 100 papers. His presentation topic is improving crop seed quality and stress resilience through environmentally sustainable technologies. Yes, Dr. Gerard Lebner, please.
Yes, thank you very much. If everything is working, I'm pleased to be a speaker at this great conference. I have some contacts to Turkey and also some collaborators there. What you see on this slide here is our founders building at Royal Holloway, University of London. It's from the Victorian times. It's like a Bill Gates foundation, the founder made, and it was in the Victorian times a women's only college. So very provocative. We are not in this building, but in a more modern or ugly looking concrete building and with nice laboratories to do our research. So this is located west of London, here close to Syngenta, which is in the region, and also the Royal Horticultural Society and some seed companies, and another seed bank, namely Q, compared to the IPK seed bank where Manuela presented just before me. The Q seed bank has more wild species, and it's also a large one. So I'm not from a seed bank. I lead a research group, and I will present our work into improving crop seed quality and stress resilience through environmental sustainable technologies. This works very well. So this is my research group. Due to the pandemic, I only have an older photograph. So some people have already left on this slide, and some are new. I just want to point out some essential points of my lab. One is that we have different expertises. An example for this is Tina Steinbrecher here, who is a biomaterial engineer and who also collaborates in some of our projects. I will come to these with electrical engineers to develop agri-technologies. Others, like Jake Chandler, works a lot with sugar beet. He's in a project with TWS and is mainly a transcriptome or molecular biologist. So what is our work addressing? It's addressing what we all know and have also heard again on the COP26 summit, namely that our climate is changing, and this is a very interesting publication here in Nature Climate Change, and Nature from two publications in the journal Nature Climate Change, which really tells that the climate had already changed by evidence in 2012 when it was compared to earlier times. These changes in global weather and climate are not only an increase in temperature, they also come with an increased risk of extreme weather events, and these are probably even more important for sustainable agriculture, because if a drought hits your crop at the wrong time, then it can be quite devastating, and it is also more difficult to develop technologies and breed for something which is not an increase in temperature in general, but is events which are extreme and sudden and unpredictable. For temperature, we know a lot about some genes. One includes the so-called DOG1, delay of germination 1 gene. It controls dormancy in seeds, but it also determines the window, permissive window, temperature window for germination of seeds. I will not discuss this. This is very well published, for example, in our PNAS work here in 2014, but also by others. For example, Stephen Footage showed the cycling of this gene expression in Arabidopsis. Other important regulators for these responses are hormones. And here, of course, the famous balance between abscisic acid as an inhibiting hormone for germination and as a mediator of dormancy and the germination promoting and dormancy release inducing hormone gibberellin is important. So it's both the biosynthesis, degradation, as well as the signaling for these hormones, and thereby the changes in sensitivities, by which seeds, as very sensitive environmental sensors, adjust their responses to the seasonal, but also to the prevailing immediate weather which exists in the ecosystem or the agri-ecosystem. So gibberellins, oxalic acid, and DOC1, these are for sure very important players which have a role in these responses in all seeds. There is, of course, more. There's more regulators than these three, but they are among the most important. I now want to turn to one example for this and for our research. And for this, come back to what Jake Chandler in my lab is doing in collaboration with KWS. Headquarters are in Einbeck, Germany, but KWS also has a new production plant in Turkey, and I know also the Turkish colleagues of KWS, so they have a very modern facility there. So we have collaborations with KWS into sugar beet seed quality and crop seedling performance since very long. I stayed here 2015, but it started already before when I was still in Germany. So in fact, the collaborations go back to as early as 2005, so very long time. And it is both into supporting their breeding, essentially looking at genotype biodiversity and genotype climate interactions, as well as developing physiological technologies. In case I want to present the seed priming for having high vigor and abiotic stress resilient seeds or seedlings once they're germinated. The sugar beet seed is in fact a fruit. And as you see here in this drawing, there's a dead pericarp, which is rather thick, a fruit coat, and controls a lot of the processes, what can get in and what can get out of the seed. And this surrounds the true seed here, which has a very thin seed coat, the tester, and it is a curled embryo around a nutritional tissue, the perisperm. So in seed production, commercial seed production, the goal is to improve the quality of this seed, which is then sold to the farmer to the maximum possible extent, both by breeding and by seed processing technologies, to at the end sell these pellets, this here, which then germinate fast, establish fast, and provide a maximum sugar yield in the product. We have looked into the biochemistry of seed technology and processing in sugar beets. This is, for example, this publication here in Planta. And in principle, the various processes here include from the unprocessed harvested seed, or better fruit, if we are botanically precise, that these are polished, primed, coated, pelleted, the different ways of improving them. So what is always done is polishing, getting rid of these parts here which are around the pericarp. And Tina Steinbacher in my lab, the biomaterial scientist, she has worked on the polishing process and what the basis and effects of this is, because you can polish too much, too little, and this affects seed quality. And then priming is conducted, and at the end it's coated, and these pellets then are sold, and they provide rapid germination and seedling emergence, or if you want this to hear this term, it has a high vigor, namely a rapid and strong downward growth of the root, and then a rapid and strong upwards growth of the shoot here. And this is the most vulnerable stage in the establishment. So you can imagine that when a bad weather, like a drought, hits the seedlings in this state, you can have the best possible genotype, it will not produce any yield. So proper establishment, protecting the very vulnerable early stages here, is really, really crucial in any production. And this can be improved not only by breeding, but also by priming. And with priming, you speed it up. And this is a means of stress avoidance, because of course, if you have a establishment within, let's say, 1 week compared to 3 weeks, the chances that bad weather hit them in very vulnerable stages are very much reduced. Tina has devices for biomechanics, and she also worked with wheat grains. So we're not working only in sugar beet, but I use the sugar beet as a prime example for how we tackle issues like bad weather. Examples are here: flooding, waterlogging, drought and heat damage, but also increasingly frost damage. The point is that due to the warming, farmers start drilling earlier. In sugar beet, this is compared to a few decades ago, this is about 1 month earlier. And then of course, if it's earlier and the seedlings are hit by frost in the early spring, then there is also a problem. So it's a lot of different types of stresses, and you can have combinations of stresses which then form a special stress, which is not just the addition of these two stresses, but a stress in itself. I will stay a bit with chilling and cold, because we found something interesting in sugar beet, which also exists for brassica, which can be prevented by priming. But before I do this, a few words about sugar beet, and this is something important also for breeding sugar beet varieties which grow well in regions. This is what we do in the moment in collaboration with Syngenta, with PWS, not the breeding, but supporting them in assays, also for regions which have more drought or which are prone to salinity. And for salinity, sugar beet is pretty good crop, because it is in fact related to the maritima and the sea beet, which is adapted to salinity. But then there is also further relatives which have a very hard pericarp, this structure around the seed, and this hard pericarp in these relatives is a means of physiological dormancy. And so the question arises whether cultivated sugar beet is either non-dormant, or does it have some physiological dormancy? And there is something which we found in our work in collaboration with TWS, namely that one of the seed lots, I simply call this seed lot here 310, showed no germination or very low percentage of final germination at very cold temperatures, at 5 degrees here. The low germination went only up to 20% compared to 10 degrees, where it goes up to 100%. And so we thought that maybe this 80% here, which does not germinate at 5 degrees, may be dormant. And this is what we looked into. What is also clear already for what is the priming process, if you prime it, then you do not have these effects. So the priming as such prevents it. And it makes sense, as it is known that in sugar beet, priming is associated by a significant increase in the gibberellin content. But before I come to the priming, let's look at this secondary dormancy induction, what this indeed is, which is induced by the chilling. So if you take this 80% of seeds here and move them to another condition where you add gibberellins, you can completely break it. So this is already all you. If you move it to 10 degrees, it won't germinate further. So it is a true dormancy, which is induced. It's not just an inhibition by the cold temperature. And if you add gibberellins, then you can break it, which is an indication that this is indeed physiological dormancy. And this physiological dormancy it was also evident from emergence assays, but in the emergence assays you only see the seedling emergence. You do not know whether it's on this stage or on this stage. We verified that it is at this stage. This is the line 310, which I focus on. We also screened a couple of different other seed lots, and they do not show this phenomenon, or many do not show this phenomenon. For example, the seed lot 257 germinates 100% also at 5 degrees. If you look at seedlings, on the other hand, you compare 310 with 257, their growth rate, as fast as they grow, either the root or the shoot, is no difference between 310 and 257 once it has germinated. So this problem which occurs here seems to be a really secondary dormancy. If it's secondary dormancy and gibberellins can break it, you would expect that it is affected in the ABA metabolism and signaling also. And this is in fact the case. Here's the time and here's the ABA content of this sugar beet seeds at 3110, either at 10 degrees, where it goes down and it germinates them, or at 5 degrees, where it initially goes down, probably due to this 20% which germinate, but then it goes up and up and up, and during secondary dormancy, induction stays high. So if it goes up, it's most likely that some of these genes are here affected accordingly, and you can test this by either doing qPCR or what we and we did this, and we also did a transcriptome analysis. So for the transcriptome analysis, we have a special sampling scheme here, which I do not have time to get into. But for example, this gene here, the initial step in the committed step in ABA biosynthesis is showing this gene increases in secondary dormancy and it decreases in the germinating situation 10 degree. The opposite is true for the ABA degrading enzyme, which causes this rapid decline. It increases in 10 degree and it decreases in the secondary dormancy induction. So ABA is clearly one of the factors in this situation. There are many more in the transcriptome, and due to the time, I restrict myself to another player, and this is the ACC oxidases, which are producing the hormone ethylene, and ethylene has some role also in germination, namely that it stimulates it. And what you can see is that it increases here in the 10 degree and that they remain low, the expression in the 5 degree. And so this fits very well with dormancy induction. But this was dormancy, but what about priming? I mentioned that priming technology can help dealing with the stressors, and just run fast through what it is, because I assume that most of you know what it is. Usually you have a water uptake in a seed in a triphasic manner. First imbibition, then second process it stays similar, and in this stage here there is metabolic activation, and then there is a third increase in water uptake which is connected to the elongation growth. And if you now prime seeds, what you do is you prevent that it can go from the activation state to the growth state here, in limiting the amount of water you add. And this then leads to the situation that after you dried it back, you can store it. And if you then place it back in the field, it's much faster, because essentially this activation state here is already done. The metabolism is already expressed and there to provide fast establishment. Okay, so this is how it looks in nature for sugar beet pellets. This is the pellet state. This is the state where it germinates, the pellet breaks, and the radical protrudes. And then here's in the primed stage, 10 degree, it would already be in the seedling stage already here, whereas in the control unprimed, it would be less advanced. So the process of establishment is very much promoted in primed seeds. Okay, this we have seen before also. ABA decreases in seeds and germination occurs for sugar beet. And for sugar beet, you have to distinguish between the true seed and then the dead pericarp, which is surrounding it. And ABA measurements in primed seeds show that the decrease is faster of ABA degradation. Also, ethylene production is pretty late during sugar beet germination and early establishment. And if you compare the expression of the ACC oxidase transcripts between primed seeds and control seeds, the ACC oxidases are already, the transcripts are already elevated in the dry state. And if you imbibe them, it even goes up further. So in a way, it's not simply that during priming there's only the metabolic processes activated. They are in some cases, I would say, over-activated. So you have more of these here than you would have in a control seed at the end. The disadvantage of priming is that seed storage may be compromised. And this is also what you see. If you age primed seeds, then this activation becomes lower. So there is a faster decrease in the promoting factors. And this aging period, therefore, is a critical one. And one can think about technologies which would do both: prime and activate seeds, remove dormancy, and retain the untreated storability. And we, in fact, developed such a technology, or we think that it is such a technology which is valid in many different species. So I'm not talking about sugar beet now, but about a seed technology which we developed, and we call it ePower. We have submitted this as a patent. So the ePower seed technology uses gas plasma activated water. I come to what this is in a moment. It offers a practical and cost-effective high throughput method by applying this gas plasma activated water to commercial crop seeds, resulting in dormancy release, improved seed vigor, while maintaining aging tolerance and therefore storability. As I said, it's not yet tested in sugar beet, but it's tested in many other species, and I will present some of these results here. So what is plasma? Plasma is produced by the sun, for example. It's called the fourth state of matter. So you would have solids, liquids, gases, and then plasma. And plasma, for example, occurs if you have lightning here. And as you know, during lightning, also nitrogen can be fixed from the atmosphere. But the plasma is, in principle, a mixture of reactive oxygen species, reactive nitrogen species, other active species, which are formed, and it is a very intensive chemistry which is going on. We are here, of course, not talking about the sun, which produces plasma and has a surface temperature of 5,800 Kelvin. We're talking about a non-thermal, or so-called cold atmospheric plus pressure gas plasma, which means the gas plasma which is formed under normal pressure and at normal temperatures as we have them in the lab. And this is what we did together with our collaborator at Loughborough University and electrical engineer Felipe Itza. And this is the device for this. I come back to the device in a minute. First, I say what I think the importance of gas plasma technology is. It can be the basis of a sustainable agriculture. In this review here in Trends of Biotechnology, it was proposed to increase germination and yield, provide nitrate fertilization, reduce bacterial levels at harvest, degrade, because it's radicals, pesticides, maybe there are remains of residues of pesticides. It may remove pests and mycotoxins, It can be used for sterilization of food. It can reduce food waste by having the post-harvest resilience higher, and liquids can be used for decontamination. So a lot of possibilities were proposed for using these technology to do another type of agriculture. There's also a cost action if someone is interested joining this field, where we also attend. We are not the leaders there. Okay, so we have this collaboration with Felipe in 3 projects funded by the BBSRC, and I want to show some results. I hope this works, because this is a movie where I usually have to press here. It doesn't work, or is it working if I press again? No, it doesn't. Anyhow, this would generate lightning if I would show the moving here, and this lightning occurs between 2 electrodes here in this reactor, and these electrodes they are producing high voltage and apply this to a carrier gas, which is usually simply air, compressed air, which flows through it. And then in this situation, gas plasma is produced, and then this apparatus is bubbled through distilled water. And so the water becomes gas plasma activated water, and gas plasma activated water is sometimes also called by us magic water. What happens in this gas plasma activated water is that some species are formed, and we measured them. So this is the primary chemical reactions. There's, for example, hydroxyl radicals here in red. There is a bit of H2O2. There is nitrate formed here in blue, and you can use air, as in this case, but you could also use a helium-oxygen mixture. Then, of course, the nitrate is low because there's very little or no nitrogen input, but hydroxyl radical formation is going on. There's not only the primary reactions, but there are also secondary reactions. So if you stop the reactor, chemistry goes on, and the chemistry maintains a level of nitrate production, a level of hydroxyl radical production, and also H2O2, and many more. So this is only some of the ingredients. Hydroxyl radicals are very aggressive molecules, but they are also very, very short-lived, I think femtoseconds. So as soon as they are produced, they grab any molecule next to them, react with it, and then they decay. Okay, if we now use such water here, but at 45 minutes or 60 minutes, and apply it to Arabidopsis seeds, which are dormant, then what happens is that the dormancy is broken and they germinate. And remember from earlier presentation part, the dormancy release may be related to gibberellin synthesis and ABA degradation, and dormancy maintenance by ABA synthesis. So this switch between no germination and germination, the dormancy release, can be mediated by treating with plasma activated water. Here you have fresh mature seeds of the wild type, germinates only 15%, and if you activate it with air gas plasma activated water, it germinates almost fully. Nitrate is also active. If you do this with after ripened seed, you have some increase, but of course the effects are lower. If you use so-called SIP7 or 7A2 seeds, these are seeds which are unable to degrade ABA because the enzyme is missing. The activation is in part there, but it is not as severe as with the wild-type seeds, suggesting that ABA indeed plays a role. And I won't go into details with the helium-oxygen plasma. What we then did is we measured the transcript expression of different enzymes which may be involved. And yes, at 6 hours and at 24 hours, I may stay with the 24 hours. Here is XTH, a cell wall degrading enzyme, a cell wall modifying enzyme, and expansins, which are also required for germination as they modify cell walls. Also, these transcripts are increasing, and the early times, even already after 6 hours, the ABA degrading enzyme is produced, and also GA3 oxidases. The same is then true here with helium-oxygen plasma. So these gas plasma activated water mediates its effects, its germination promoting and dormancy releasing effect, in part at least by inducing these gene expressions by the ingredients it contains. And here you always have to compare it with the nitrate control, which does something, but not everything. As you see, here is no nitrate induced in the helium-oxygen plasma, but it still releases the dormancy. What we also did, or Tina did, is measured the weakening of the endosperm here, and the result is, yes, gas plasma activated water promotes the weakening, but this weakening promotion is not simply due by gene induction, it's also by direct action. I skipped the slides here due to time. We looked at different mutants, and these mutants are for the different pathways which may be involved in this dormancy release. And we always compared the plasma with the nitrate at different doses and different nitrate doses. And these different mutants include the SIP7-7A, then LPA8, which is for a nitrate signaling pathway, PRT6, which is for the enrolled pathway. And what we found in general about with these mutants is that many of these pathways are involved, but none of the pathways is simple. It's not as simple as this one pathway which is mediating the gas plasma effect. So this is a summary of this. Nitrate is involved, leading also to ABA degradation. Nitric oxide signaling leads to ABA sensitivity removal. Reactive oxygen signal leads mainly to gibberellin synthesis and to direct cell wall degradation. So we think this is a very interesting technology, and it is suggested also in the chat that I should tell a bit in addition about pelleting. So usually priming is done after polishing, and then the pelleting is done. Some companies have special procedures there and I can't go into the special procedures. In any way, the pelleting after the polishing is adding material to it, and you can add something into these pellets, which also helps to improve the germination, in addition to the priming of the seed, which is done before this. These additions today, and I won't talk about this, I simply will present it, can be beneficial microbes. So if you have beneficial microbes, you can also use enzymes, hormones, allelochemical or beneficial microbes, and you add them to the seeds or to the pellets, they may help establishment also by interfering with the different hormonal pathways and thereby provide protection and also increase of vigor. So with this, I come to my last slide, which is thanking my co-workers in the lab. I presented results mainly conducted by Tina, Jake, Marta, Giles, and in collaboration with Felipe Itza. And our collaborators are listed here and include TWS as one of the collaborators for which I showed results, but also our colleagues in the Czech Republic, where we have a good collaboration for hormone analysis, and they were involved in this work too. Thank you very much for listening. I'm happy to answer questions.
Thank you very much, Dr. Gerard, for this informative and perfect presentation. I think there is one question from Dr. Manela. Dear Gerd, thank you very much for the nice and clear presentation. I wonder at which stage of commercial sugar beet seed production a priming procedure is included. Is it a separate process before pelleting or is done during pelleting? Yes, Dr. Gerard, please.
Well, I tried to mention this already in one of the slides. So in commercial sugar beet production, priming is done first and then pelleting is done, but some companies may have also combined the processes. But even if it's combined in biochemical terms, the priming comes first and then the pelleting.
Okay, thank you. The other question from Pakistan, from Faryal Ali. Dear Dr. Lebner, is there a way to the cellular degradation that occurs during aging in seeds with priming methods? If so, do you think this improvement comes from a change in the DNA?
Yeah, I mentioned that often primed seeds are more prone to aging, and this aging also includes DNA damage. Depending on the genotype, then the DNA damage can be repaired easily during inhibition, but some genotypes may be better there than others, depending on the repair enzymes and repair systems which they have either already in the dry seed or induced during the priming, like antioxidants, ascorbat, for example, which helps as an antioxidant to prevent further damage and supports that the repairs are done. Yeah, DNA, changes in DNA and improvements there are very important, yes.
Okay. Thank you, Professor Gerard, for your presentation.
Thank you for listening and asking.
We thank you. Yes, the next speaker, Professor Dr. Gregor Wellbaum. Dr. Wellbaum is a professor of plant biology at School of Plant and Environmental Science, Virginia Tech. He has obtained his doctoral degree at University of California in 1988. His researches have been focused on alternative seed testing methods that could replace paper towel tests and provide realistic assessments of seed quality and field performance. In a laboratory, he is also investing both non-destructive scanning technologies and the lab field TM system. The topic is lab field TM as a tool for predicting field emergence of diverse melon. Yes, Professor, please.
Okay, can you hear me okay?
Yes, we can hear you clearly.
Great, it's great to be with you on the other side of the world.
Thank you. We are very honored to hear that.
Well, thank you for inviting me. I wish I could be there in person, as I'm sure all the other speakers could, but given the times, it's nice that we can connect electronically. Okay, let me go ahead and just explain. I'm here in Blacksburg, Virginia, in the United States, at a university called the Virginia Polytechnic Institute and State University. Here is my contact information on this first slide. If anybody wants to contact me and talk about seed research or any of the topics I'm talking about today, please feel free to do so, because I know it's kind of awkward to ask questions in this format, not being face-to-face. This is a picture of my building on the bottom, my laboratory, which is a little bit crowded. And that's me. I'm kind of unique that my family owns a farm and we're actually involved in production agriculture. So that's the picture on the left right here is actually me standing in a soybean field in the United States, in the state of Ohio. So as you were told, the topic today is going to be about the comparison of gradient table, field, and paper towel germination tests. We used melon for this particular experiment. And at the beginning, I'd like to acknowledge the people that contributed to this. Stephanie Baum, who is related to me, did a lot of the field research you're going to see today. Michael Beal, one of my graduate students, did a lot of the statistical analysis for us. And Imo Fu also played an important role in this project as well. I just wanted to mention some other research going on in my laboratory right now. Obviously, I can only talk about one project, but we are involved in a number of other projects studying resistance and treatments to control Acetovorax citriuli, which is known as watermelon fruit blotch disease, is a major project that we have been involved with for many, many years in collaboration with Saul Birdman in Israel, Ron Walcott at the University of Georgia, and my colleague Bingyu Zhao at Virginia Tech. So that's probably my biggest research project, and I think we're making good progress on that particular project. We've been at it for a long time, so we should. I have another project a student just finished about passive solar systems to heat low tunnels for season extension of small fruit and vegetable production. And in a project that's very similar to what Gerhard was just talking about with the sugar beets, was we have a project to improve the establishment of vegetable soybean using biological seed treatments. And a lot of the things that Gerhard talked about in his presentation for sugar beets also apply to our work with soybeans. So the reason we conducted the experiment I'm going to talk about today was that we feel it's important to have more realistic seed germination tests that better predict field emergence, because with global warming, global climate change, seeds aren't always emerging and you're not establishing your crops under ideal conditions for the area that they're grown in. So there's more environmental variation than there used to be. And the question is how can we better assess the performance of seeds to predict how they will do in these different environments. The other thing is that a lot of the seed testing procedures used by both ISTA and here in the United States by AOSA are based on paper towel tests. So if you're working in a laboratory testing thousands and thousands of seed lots a year, you're using a lot of paper, and paper is a renewable resource, but it's not the most sustainable system. So we're looking at ways to become more sustainable in the way that we test seeds. Today I'm going to be talking about a tool for seed testing that we developed called LabField, which is basically a thermogradient table that contains soil, and it's been modified with vertical elements welded onto the base of the plate to control soil temperature, so you can mimic field conditions with a variety of different soil temperatures in one experiment in a laboratory. So it's called LabField because you're simulating field conditions in a laboratory. And then we decided to use Cucumis milo, which is a species that I work extensively with, cucurbits in general, for my research project. And that, you could do the research we did today with any species, but since we work so frequently with Cucumis milo melon, that is why we chose to use that today. So this is a picture, the picture of the melon. Here in the US, we grow a lot of netted melons. A picture of Athena, which is a popular cultivar here in the United States, is shown in this picture here. We compared the field, laboratory, and paper towel germination performance of 5 genotypes in this experiment: Minnesota Midget, Chardonnay, Hale's Best Jumbo, Edisto, and Athena. These are diverse cultivars with very different characteristics. They were all purchased from the same seed company. I'm sorry that got cut over as the picture shifted a little bit, but by a nearby seed company here in the United States. Now let's talk a little bit about the tests that we used. We planted the seeds of all 5 cultivars in a field 2.5 centimeters deep on clay loam soil that was watered daily. We didn't want water to be a mitigating factor. The soil temperature was 26.3 centigrade, and the date that they were established in the field was the 28th of June 2020. Emergence was counted twice daily. So this was during the pandemic, during the beginning of the pandemic, when we couldn't work in our laboratory. And one of the reasons I designed this experiment is we could do it in the field, and individuals, not groups of people, but individuals could do laboratory experiments. So this was from actually last year during the height of the pandemic. The other treatment was the AOSA, and if you're not familiar with ASA, it's very similar to ISTA, towel germination treatments, which for melon is alternating 20-30 degrees, 16 hours at 20, 8 hours at 30, in the dark on 4 sheets of paper towels, 1 dry, 3 moist, with a first count at 4 days and a final count at 10 days. So that's kind of the standard industry procedure for assessing germination performance of seeds. And then the final treatment was the LabField simulated field environment using ProMix potting mix. We adjusted the table to 4 different temperatures: 12.5, 17.5, 25, and 31 degrees C. We hand watered the plots, and emergence was counted twice daily, the same as the field plot. As far as statistics is concerned, paper towel germination was tested as 3 replicates of 50 seeds each in an incubator. Data was collected on root lengths after 4 days. When we did the early count, we wanted to get a little extra data, so we measured the root lengths at that time. For the field and gradient table plantings, we had 3 reps of 20 seeds each in a randomized complete block design. Emergence was counted twice daily. The mean time to germination was calculated using the summation equation, basically counting the seeds for each day and multiplied by the total number of seeds that germinated for each time. And then ANOVA was performed on log transform time data. and arcsine transform percentage data using the computer program R. Mean time to germination was not calculated for treatments where there was less than 25% germination because of an insufficient portion of the population germinated to really sample that population. So if you have less than 25 seeds, you really don't know what the characteristics of that population are. And we figured it wasn't fair to compare that small number of plots or emerging seeds to other populations where we could sample more of the population with greater germination. A mean separation of the ANOVA was by TOHUMCULUK's mean separation test. This is a picture of the field plots. You can see it's quite simple, just soil covered uniformly as we could. Each one of these lanes is a replication of the seeds. And then we counted cotyledon emergence as germination in this test. This is the paper towel test, again, similar to the ISTA testing procedures, where we use sheets of 50 seeds, rolled the paper towels, put them upright in an incubator, and then went back at 4 days, counted emergence, measured root lengths, and then did a final count after 10 days. A very similar procedure used around the world. You can see that it uses a lot of paper towels if you do a lot of this type of testing. So this is what the paper towels look like when they're unfolded. And you can see the root length and the little seedlings growing on the paper towels. And this is a picture of the thermogradient table. This is one from Oregon State University. A lot of seed labs around the world use these devices to test seed germination. There are all electronic models of these available now that electronically cool and heat the table. We're using a little bit of an older design that circulates antifreeze at both ends of the table to set up the gradient passively between the two extremes. So the circulating baths underneath here are circulating, pumping chilled solution through tubes welded on the bottom of an electric plate. And this will become obvious in the future slides. This is a conventional gradient table or a classic gradient table without the gussets. Usually experiments are done on Petri dishes, or in this case, sandwich boxes with blotter paper. Some areas actually put the rolled paper towels directly on the table, covering them so they don't dry out to test germination as well. Now in our design, we welded these vertical elements, gussets we call them, onto the base of the tray so we can add soil. And the temperature is conducted through these fins or gussets so that the soil is controlled at the gradient of temperatures that on a classic table is only represented on the base plate of the design. So this gives us some vertical temperature control, allowing us to control the temperature that the seeds are exposed to for the germination test. So you can adjust these two baths for any range of temperatures. We can actually freeze ice on this device, or we can go up to 45, 50 degrees if that's necessary, and test germination at a range of different soil temperatures in one experiment in a laboratory under very controlled conditions. So this is the result of an experiment. You can see where seeds at the lower temperature at this end of the table are inhibited, where seeds at the warmer end of the table germinate very rapidly. And then you can calculate mean time to germination or germination rates and germination percentages for each of these temperatures and get a lot of data. The tables can also be installed with LED lights, so you have a light environment, which makes it like a mini growth chamber, except that the air temperature isn't controlled. Only the soil temperatures are controlled at different temperatures, depending on how you set it up. So each channel or lane here is a different temperature. This is an earlier experiment. It's not our experiment, but you can see in this experiment, there's no germination at the low temperatures, some germination at the intermediate temperatures, and then rapid germination at the high temperatures, as you'd expect. So if you're doing experiments with seed priming or seed coating, you can test the effects of those different treatments at different temperatures and determine optimum temperatures. If you're working with native species or seeds that haven't been experimented with a lot, you can determine the optimum temperature for different crops or wild native species of plants. There's a lot of versatile uses for this table. Also, people are starting to use this to study soil properties. So if you want to study microbial activity at different soil temperatures with different amendments, different fertilizers, that can be done as well without even growing any plants at all. This is an example of that. This is a company in California that measures gas exchange from soils to see how much of different nitric oxide and different gases are produced by soils. So you can set up their equipment on top of the lab field table and measured how much gas is evolved at different temperatures and different types of soils. Of course, you can replace the soils with any soil that you want to work with. You can also inoculate the soil with different microbes and just see what the effects are on gas exchange as an example of one of the many uses for this device. So here's another picture of a table. So this is our experiment that I'm describing today, and we'll be showing you data for. This is the melon study compared to the paper towel and field emergence. We wanted to see how accurately this could predict field emergence and compare it to the paper towel test, which is the industry standard. So the seeds were seeded in lanes very close there because obviously we're not growing them to maturity. We're just interested in emergence data. Each of these lanes represents a different temperature. Again, we had 4 different temperatures, and then they're uniformly covered with 2.5 centimeters of soil watered by hand daily. So this is after watering and the soil is added, and you can see right here a tiny seedling is emerging. So we would come through twice a day and just pull off the cotyledons of the seedlings that emerged to keep track of them, recorded the time and date, and then compared these data to the field data. There's the watering by hand. This does have a drainage system. So if you accidentally overwater a lane, the excess water will flow out. And for this experiment, we were using potting media, ProMix, that is very well drained and doesn't retain moisture to a great extent. We also took data on the soil temperature, and that's how we know what the soil temperatures that I reported were. There is some variation from the top to the bottom of the soil profile. So we averaged the top, middle, and bottom temperatures, which was typically 2 degrees centigrade or less. And of course, the seeds are kind of in the middle, so that's the temperature that they experienced. But again, there is some variation, but it's manageable. It's not too great. And if you record this, you can accurately determine the temperature. From one side to the other, the temperature differences are minimal. So the temperatures are consistent across the table. Just with depth, you see some variation as would be expected. You can also put data loggers, watchdog data loggers at different profiles in the soil to do this automatically so you don't have to measure manually temperatures every day. So if you put a data logger in here, it will log, and then you can take it out and see what the temperatures the seeds are exposed to at different profiles of the soil. So this is a rather busy slide, but this is an example of the statistical analysis. I don't want to go through all the statistics, but this is the arc-sign transformed germination data. And if we can skip down here to the bottom, you can see that we did see differences between, if we look at the probability here, between cultivars, treatments, and there was a significant cultivar by treatment interaction. If we look at one particular test here, this is the test of between cultivars across all different tests. You can see that the cultivars perform differently, as you would expect, because they were quite different, diverse cultivars of different seed quality, as it turned out. I don't think that these differences are real genetic differences. They're more a reflection of seed quality. For example, even though we purchased them all from the same seed company, the seed quality of the Minnesota midget was very, very poor. They hardly germinated at all, even though they were commercial seeds. And then this is an example of a comparison between field paper towel and one temperature. Because of our model, we couldn't compare all temperatures in the same model. So in a temperature of field, paper towel, and temperature 1, you can see that the field and the paper towel weren't significantly different, which was a little bit surprising, but the gradient table was. And the point is that it depends on which temperature you're comparing, obviously. If you're comparing a temperature of 10 degrees C and the field soils are 25 degrees C, you're not going to get a good match. So we also did these same comparisons for mean time to germination, and I will just, rather than show you where those data came from, as I've attempted to do here, I will just summarize the actual data in a table. So this is a comparison of field, gradient table, and paper towel tests for germination percentage. This is an arc-sine transform. These are the actual percentages. They were analyzed on arc-sine transform data, but I'm showing the differences here on the real data because it allows you to visualize the differences more easily. But you can see these represent the 4 temperatures on the gradient table, which were 12.5, 17.5, 25, and 31. Now we know from our field data that the actual field temperature was 25 degrees C. This third lane here is kind of indicative, the best one to compare to the field results. But you can see that the field, it seems kind of strange to me, but the field produced the highest germination percentages in our study. And I think it was because we did this study in the middle of the summer when soil temperatures were quite warm. The conditions were very favorable because we watered daily, so there was no moisture stress on the seeds. It was almost ideal, so the seeds germinated very well in the soil. A production problem for us is commonly planting this crop in cooler soils in the spring, where they typically don't germinate as well as they would in a laboratory. So a more common problem for the industry is to underestimate field performance with the paper towel test, because the paper towel test is done under optimal conditions, where field conditions usually are not, but this is a little bit of an atypical situation. And if you look here at this lane, you would see that for certain cultivars like Chardonnay here, the gradient table did a nice job of predicting field emergence at 25 degrees C, but less so for other cultivars, which is kind of interesting. So Athena was well predicted, Hale's Best Jumbo was well predicted, Chardonnay, but Minnesota Midget, the poor seed quality cultivar, and Edisto did not perform as well in the gradient table as they did in the field, which was a little bit of a surprise. And you can see the paper towel test is pretty much similar. Poor prediction of Minnesota Midget, but the other 4 cultivars were predicted quite well, and that's why there was no statistical difference between those tests in our analysis, ANOVA analysis. As far as the mean times to germination are concerned, you can see that in the field, Minnesota Midget, Chardonnay, Minnesota Midget was very slow, as you would expect, since it was poor quality seed. The other cultivars germinated much faster. These are days, data in days. So the mean time to germination for Chardonnay was 3.4 days. And you can see there are differences here between the cultivars. As a comparison, we did the paper towel 4-day root length because we couldn't really calculate mean time to germination unless we opened up the paper towels and counted every day, which we did not do. But you can see there's kind of an inverse relationship. The The slow germinating seeds had short root lengths, is what you would expect. And the seed lots that germinated fast had much longer root lengths. And then again, with the gradient table, if you pick 25 degrees, you can see there is pretty good correlation, or some correlation between that temperature and the field experiment, which mimicked that temperature quite well. So some conclusions from all of this. Even though seeds were purchased from a commercial supplier, there were major differences in seed quality. Both Edisto and Minnesota Midget failed to germinate to the minimum standards of the U.S. federal seed law across all of our tests, which is concerning because these were sold as fresh seeds from a commercial supplier, yet they performed very, very poorly. Paper towel test reliably predicted field emergence. This was possibly due to planting seeds in warm soil during the summer, when soil test temperatures were very similar to laboratory environment. The lab field tended to underestimate field emergence, especially at below 25 degrees centigrade, which is understandable because you're testing seeds at lower than they were exposed in the field. Lab field provided more information about performance at temperatures other than the optimum, which could help predict performance at suboptimal temperatures. So one of the outcomes from this is if you do a lab experiment using lab field over a variety of temperatures, you're generating a lot more data, and that data can be applied to different conditions that you may not be exposing them to. So you're just getting a lot more data when you do a lab field experiment over a variety of temperatures in one single experiment. Both Edisto and Midget, Minnesota Midget, germinated more slowly than other cultivars, a sign they were further along the death curve, or they had been aged seeds at somewhere in their production. Seeds germinated more slowly with decreasing temperature on the lab field table, which may be helpful for predicting emergence in cold soils and identifying cultivars. And seed lots that perform better under cold conditions, such as Chardonnay and Athena. We've done a bunch of this work with melon with my colleague in Israel, Menachem Edelstein, and we've actually shown that there is significant genetic diversity with regard to the base temperature of melon. So it is possible to find cultivars and genotypes of melon that do perform better based on their genetics at lower temperatures than other temperatures. Hale's Best Jumbo performed well at warm temperatures, but not at cold temperatures, suggesting there may be variation among the cultivars tested in this study, but of course a lot more work would be needed, and we didn't actually calculate a base temperature in these experiments. 4-day root lengths had value predicting germination performance and yielded results as an inverse of the mean time to germination data. So if you don't have the ability to measure mean time to germination, you can take that 4-day root length count and it'll give you an approximation of the mean time to germination in one measured experiment. So I believe that is my last slide. Thank you for your attention, and I'll be happy to entertain any questions that anybody has.
Thank you very much, Dr. Wellbaum. Thank you very much for this excellent and informative presentation. It was very nice to listen to you. Yes, is there any question? Yes, I think there is from Gerard Lebner, Dr. Lebner. Yes, Lebner.
Sorry, can you hear me?
Yeah.
Yes.
Yes, yeah. I just started to type. What I found interesting is your comment, Greg, about the genetic basis of base temperature, where you found variation. This is very interesting, I think. Have you any idea whether someone has got into some genes which are related to this, or is this not as far as it could be?
Yeah, in our study, we found that there were differences in oxygen. So we related this to the ability of certain seed coats to pass more oxygen to the embryo. So the mechanism behind melon, at least the genotypes that we studied in Israel, were more porous seed coats, were at least correlated. Now, we didn't look at genes, but I imagine that in the world has done that. I just can't cite a reference for that.
Yeah, that's interesting with the oxygen. It's very interesting, yes.
Yeah, it's a paper in Crop Science we published about 10 years ago.
Yeah, thank you.
Any questions? I think so. Thank you very much again, Dr. Gregory. Thank you very much.
Thank you for inviting me.
It was a very honor to us. Thank you. The next speaker, I had the honor of working with him and learned a lot in a short time. Professor Alan George Taylor. Dr. Taylor is a professor at Horticulture Section, School of Integrative Plant Science, Cornell University. He has obtained his doctoral degree at Oklahoma State University in 1981. He has been working as a professor of seed science and technology, Cornell Agritech, since 1997. His research interest is mainly oriented in seed technology and seed enhancements. In these regards, he had many scientific talks and awards. Furthermore, he has contributed to relevant seed science with more than 100 scientific publications. Yes, doctor, please. Can you hear me, Dr. Tyler, Professor Tyler?
Dr. Taylor, if you hear us, could you please turn on your microphone from the button at the bottom of the page? Okay, just click the microphone icon.
He's writing. Mikrofon icon yok diyor ama.
Hello, can you hear me now?
Yes, we can hear.
Oh, thank goodness. Little technical difficulty here in New York. Okay, well, it's a really an honor to be here, and especially being introduced by my old friend and colleague Ertan, who spent some time in my laboratory and doing some interesting work on stress physiology at the time. What I would like to spend some time with you today is to talk about more of the practical aspects and some seed treatment and coating technologies and exploiting those as delivery systems for crop seeds.
Dr. Taylor.
Yes.
You can also turn on your camera.
Okay. There we go.
Not yet. Yes.
Okay. And
yes, it's okay now. Thank you.
Okay, great. Thank you. Thank you for clicking all the right buttons here. Let's start off with kind of an agronomic life cycle of seeds, where the seeds are first produced and harvested, and then all the conditioning, sizing steps. So after the seeds are dried, then they can be handled by the seed companies and the techniques which call seed enhancements, which are the seed treatments and coatings. We could also involve seed priming in this, and we had an earlier speaker, Gephardt, speaking on that. I'm not going to cover that, but really focus in the seed enhancements on the seed treatments and seed coatings. There can be a period of time for the storage of those seeds, germination, and if needed, dormancy testing, and finally sowing of the seeds for the crop production. So let's kind of take apart this definition of seed enhancements. And this was from a review article from some of the people in our multi-state project, and Greg Wellbaum was a member of that and still is in the United States. So this was published in Seed Science and Research in 1998, and it's pretty much has withstood the definition over time. So we have different elements that I've highlighted in colors. The seed enhancements are post-harvest methods that improve germination or seedling growth or facilitate the delivery of seeds. And 3, other materials required at time of sowing. So we can take those apart. Those in blue, the performance, the improvement of germination would be our biostimulant seed treatments. We'll talk about that. Seed priming is also in that particular category. Facilitating the delivery of seeds is where we get into the seed coating technologies. And then the delivery of other materials is our seed treatments, and those can be chemical, biochemical, or biological in nature. So we have a tremendous capability on having access to the seeds during this stage where we can manipulate their future germination and seedling growth potential. I'm going to cover several things with you in this presentation. We're going to talk about seed coating equipment and the seed coatings, do some comparison with some of these technologies, then talk about the actual kind of the ingredients, the components with the actives, liquids, and solid particulates that go into a coating. Once we kind of have the basis of the equipment and the materials that would go into, then we have several case studies. As most widely known for seed treatments is in pest management. I have 2 examples with pest management. The first on onions with insect control, and the second one on post-emergence damping off, and using hemp as our example with that. Then going into biostimulants, and then also seed treatments and seed coatings to overcome abiotic stress, in particular drought stress. So we had a review article that was published in a special issue of Modern Seed Technology that I was the editor on last year. And here we're looking at the key pieces of equipment. These are not the only pieces of equipment, but major pieces of equipment for seed treatment and seed coating application. On the upper left is the dry powder applicator. This would be done with materials such as rhizobium for nitrogen fixation. This can be done right on farm, basically, as you're just applying the dry powders that adhere onto the seeds. This is particularly important for biologicals, which are short-lived or may be sensitive to wetting and drying, and we'll mention that in another slide. The next, the rotary coater. This is really the kind of the primary seed piece of equipment used now for application of binders and liquid materials, but also provisions for fillers and powder materials. And then the third is the drum coater or the pelleting pan for larger buildups of materials. So we see down below what are those kind of cartoons look like of these coated seeds. The dry coating, you'll see a small particulate materials adhering right onto the seed surface. Again, no use of water. The rotary coater we can do different things with. The seed dressing. Seed dressing is really kind of as an English term. We would call that as just that the seeds are treated seeds. The film coating, the encrustment where we're putting buildup of the filler materials all the way to the full pellet. So we have quite a few technologies here and quite a bit of capability in many different technology applications for small seeded crops, large seeded crops, high value seeds, low value seeds. I mentioned on these dry powder coatings, this is a project that we're working with. We have an entomologist at our experiment station working on lawn seed, on turf seeds, the tall fescue and the Kentucky blue. They're trying to establish a biological, but the biological is very susceptible to wetting, drying, hydration, dehydration, which is lethal to them. So we're using a dry system for that. One of the companies we're working with is quite interested in field crops. So we're also testing the application of the formulations onto these field crops. Why we're seeing this red is we need to make sure that we're applying powders onto the seeds, that we're doing a uniform application. And with that, we're adding a red fluorescent tracer material so we can visualize the uniformity of the application. Many of our small-seeded vegetables, onions is a major crop for us in New York State. Here with onions, if you're familiar with the crop, small-seeded, irregular seeds, shaped seeds. The film coating adapted from the pharmaceutical industry over 20 years ago now, where the colorants and the polymers used, and those been nicely adapted for agricultural use, wide-scale use on field crop seeds as well as vegetable seeds. We see the pelleted seeds. In the case for the onion, we need to precision place those, so using planters, the standard belt planters as we called those, we can singulate a seed and then get those placed directly in the row where we want that. And then the encrusted is somewhat of a hybrid between the film coating and the pelleting. Again, going back to our review paper, is just try to look at the addition of materials, and here going from showing those 5 coating types, the dry coating, seed dressing, film coating, encrusted, and pelleting. We put this on a log scale because of the very large amounts putting on these pelleting and encrustings would be really dwarfed on a linear scale. So here we can see about 1%, 1 to 2% with the seed dressings, and then higher weight increases as we go through more and more fillers. So again, we have the capabilities, and then the question is, how much active material do you need to load onto the seed? That also determines what sort of seed coating technology one would use to achieve that. Kind of going back to the earlier one, again, the rotary coater is a quite well used, very versatile equipment for doing a number of different coating techniques. This is the unit in our laboratory. It's a rotary pan. In English terms, R6 is for 6 inches, which you would know is 15 centimeter diameter pan. There's an atomization disc. There's not a spray nozzle per se to atomize the liquids, hitting that and then dispersing that on the seeds. So it's a very rapid process. The principles used here, even in this small laboratory scale unit, can be scaled up. Those coating pans can be 1 meter in diameter. So the nice thing is everything that we're doing in our laboratory can be scaled up to by the companies on a commercial basis quite easily. So that's really a very good aspect with our coating research at Cornell. Here we're a large, very large commercial operation where there's several large film coating pans. They're kind of ganged together. This is being used on the lawn seed, a commercial lawn seed for the cool season grasses industry, as well as other crops there in the Willamette Valley. Now we're in California, and the pelleting operation of small seeded vegetable crops of using a pelleting pan. So very labor intensive of applying these powders and liquids and binders to get everything to adhere to get the large buildups of materials. So now the question is, what do we want to put on the seeds? What do we need to put on the seeds? The first thing is the left column are your active components. And there can be a number of different materials that you could apply for particular reasons. I'm going to talk about the biostimulants for abiotic stress, and I have 2 examples with the plant protectants. But inoculants can be put on, plant nutrients. You see some of the other below that, you'll see different things like SYN and B. So are they synthetic materials? Are they natural products? The BIO is, are they biologicals? In the United States, we have a major emphasis towards organic production. So those are in green with the OR. Now, not all materials, if they're biostimulant, means they're organic, but there are examples of materials that are organically approved. The middle column are the liquids. Water is the universal solvent when it comes to dispersing materials and spraying onto seeds. Organic solvents are not used. Everything must be delivered in water. So we have colorants, different adjuvants, and a number of different binders. And some of those can be organically approved as well. The far right column are the solid particulates. Now we can have binders that are also as if small particulate binder. So what does that mean? And so all you're doing is you have to spray water onto the seeds, and the binders, the adhesives, become hydrated with the water. So again, it kind of simplifies that. And then there's a number of inert materials: diatomaceous earth, limestone, bentonite, and you can see many of those listed there. Most of those are organic. They're just mined materials from the earth, ground to a particular particle size, and then used. So going back to the active, let's say you have a plant protectant which is organically approved. But if you're going to do that and you want to have an organically approved seed treatment with the coating, that means your liquids and your solid particulates must also be using organically approved materials. So it's not just the active material only. You have to consider the whole active and the coating components that go into this to make sure that you would comply with being organically certified. So now we're going to go into a case study. Seed treatments, insecticides in the U.S., in other parts of the world, and particularly Europe, the neonicotinoids, there's a very dark cloud over the use of those materials. There's a moratorium on their use in Europe. In the U.S., we're seeing more and more environmental scrutiny on their use as a seed treatment. The primary concern is with pollinators. They've been implicated in colony collapse order, and some of the dust-off problems from the coated seeds can be a problem of dispersing those materials. So that is a major concern for the use of this class of materials, mostly used as seed treatments. The most commonly used seed treatment insecticide is the neonicotinoids. Another factor is resistance management. There's two major materials, thiamethoxam and clothianidin. Thiamethoxam is actually biochemically converted to clothianidin in the plant. So basically you're looking at the same chemistry, and anytime you use the same chemistry for pest management over and over again, there's just a probability for resistance to occur. So the need really is we need to look at alternate chemistries for that early season pest management, get away from some of the issues, particularly these environmental issues with the neonicotinoids. So we kind of go back in time now to, and this is onion plantings when I arrived in the 80s, where everything for pest management, the infra materials are applied as a drench. So in other words, as the seeds are being planted, the insecticide, fungicides are all mixed up in that big tank that you see in front of the tractor, and then as the seeds are planted, the liquids are going ahead and applied at the same time of the sowing. The growers for the precision placement are already used to a pelleted seed, and so we can see the cross-section of an onion pellet. There's ample room there for what we call pellet loading. Pellet loading is the application of the seed treatment insecticide and fungicide combinations right in the pelleting operation. Just to talk a little bit on again, back to coating technologies, there are several companies, and these are in the U.S. companies, that have their different proprietary pelleting materials. But I want to show the different types based on how those pellets fracture. We're very careful, we're very concerned that if you encapsulate the seed with an onion pellet or any sort of a pelleting material, that pellet does not hamper the germination, either delay the germination or even prevent the germination. So we really want the pellet to be the delivery system, but then get out of the way. So there's two types. Those that are tan are we call a split coat, where you can see kind of a fracture where the seeds are visibly, after they've taken up water, the pellet has taken up water, so the seeds can germinate. The hybrid is more of a fracturing in multiple places, so we call that a hybrid, which are those, the blue-coated ones on the right-hand side. So those are the two primary type of pelleting type of breakdowns. Our concern is if we're putting seed treatment insecticides and fungicides, and we have these different type of pellet structures, breakdown properties, would that affect the pest management? So this is kind of a chronology of kind of the evolution of insecticide use in upstate New York for control. Again, the time I arrived, the lure span is chlorpyrifos, which is an in-furrow material, and that's been recently has been banned, so that's no longer. The TriGuard is a seed treatment. It's our first seed treatment insecticide. I did the research on that with our vegetable entomologist at the time to get that labeled in the 1990s. Then with recent work, and since then, with another vegetable entomologist, we now have materials that contain the active ingredient spinosad. So the spinosad was our answer to a more environmentally friendly seed treatment insecticide to overcome the environmental issues that were with the neonicotinoids. The Farmore FI 500 is just a trade name, it's kind of a packaging by Syngenta that contains the spinosad. So let's talk a little bit about the spinosad. Excellent control for onion maggot and seed maggot. OMRI for us means it's organically approved. So we have a seed treatment insecticide, a biochemical material, which is organically approved. So that is very unique in itself. It has very little systemic activity. It's not taken up into the plant, so it's very well suited for below ground pest management. So we see a tremendous potential for organic crop production as a seed treatment. So I'm working with this national organization called the IR4, where our laboratory is treating seeds of snap beans, sweet corn, and peas with a spinosad to test it in multiple sites across the United States for seed maggot control. We're going to change gears now. We had earlier mention on the use of biologicals and their application. And so here we kind of look at the overview of the biological market from biofertilizers, biostimulants, and biological control products. And this next segment, this case story, will be on the biological control products. We're working in the space with biostimulants. We'll talk about that. But here we're focusing on the biologicals for disease control. And let's just kind of zoom in on what we're working with here with the biochemical. There are mineral materials, so we have organic copper and phosphites that we'll be using in this. On the microbial side, bacillus, trichoderma, and some of these have been around for many years. Basically, part of the project is we're working with the companies that are already producing these and selling these for other, say, vegetable crops, and we're going to be testing those, in this case, for hemp, which is a relatively new crop for us to grow that as an industrial crop in New York, but in many other states in the U.S. There's a lot of information here, and what I did is color-coded. If you have access to the talk later on, you can get this information. So all those in green are biological treatments. You see the trichoderma, the bacillus, and things like that, and then with the trade name of the product name. Those in red are the biochemical, the organic coppers, the phosphite materials. Those in blue are the chemical seed treatments. Anybody that has worked with seed treatments, chemical seed treatments, mefenoxam, fludeoxonil, which we know as Apron XL Maxim, these are well used in agriculture. And again, we're testing those in our particular system. So first, as we're doing work with this with the hemp crop, hemp, again, being relatively new, since there's no materials that are been labeled because of its restrictions, U.S. restrictions. We're really kind of starting at the beginning of looking at a number of materials that we're applying. So the first thing we need to do is, one is to test it for the germination, but then second is what we can do in the laboratory to kind of do the testing to test for post-emergence mortality caused by damping off, primarily from the soil pathogen Pythium. So we're looking at a time course, like Greg was talking about, with a roll towel type of a test. We're starting off with a roll towel test, and then we're using a soil, actually a sand in a field soil mixture that is sprinkled over the top of those. Hemp seeds germinate quite rapidly, so 4 days after planting, we see good germination. By 7 days, we're starting to see the disease symptoms really are taking hold. These are the non-treated seeds. After 10 days, pretty much everything is wiped out, so almost total death from that. So this gives us a good way of screening that our potential biological, chemical, or biochemical treatments before we go into the field situation. So this gives us kind of one data set to work from. On the coating part is we're looking at, we call them hemp seeds. They're actually an achene, the same thing as a sunflower. And the sunflower having a pericarp. You can see kind of the shiny nature to the seed coat. It has this kind of waxy. So when we started doing this, some of our conventional seed treatment polymers weren't really doing that good of a job for the uniformity of application over the seed surface or from seed to seed. So we reached out to Incotec at the time, and I says, do you have a coating materials, a film coating material that you would use for a waxy seed coating? And they said, well, we have something that they were using for sorghum at the time. And I says, well, we're working on hemp. They didn't know anything about hemp, but we have good relationships with these ag companies, the seed enhancement companies, and they provided this to us for our testing. So kind of lo and behold, it gives very good uniformity of application over the seed surface. Now, along with the laboratory testing, now we need to have the field testing. So through this IR4, which again is a U.S. national program for testing different pesticides, biologicals across the United States on specialty crops, and hemp would be considered as a specialty crop. We're testing at Cornell, and then we have North Dakota State head of field sites, and then Virginia Tech. Some of Greg's colleagues we're also doing testing there at Virginia Tech with our seed treatments. For sake of time, I'm just going to show you one of the Cornell studies that we had. So again, these color coding comes out to be very handy. The black bar is the control, the non-treated seeds. And in this case, all the biologicals were seeing no improvement. And this is really not just because of our Cornell study. This is in the other sites as well. So in our case, the biologicals were not working for controlling the soil-borne pathogens. We're quite enthusiastic about the ones in red, which are the biochemical treatments, and then they're as good as, in many cases, as the chemical seed treatments. So to kind of wrap this up, we see the biochemical, which are the organic copper and the phosphite, generally had good efficacy in our field tests. We're quite excited about that. The chemical seed treatments, the Apron XL Maxim did a good job, but corporate decision decided that no longer wanted to support hemp labels. And this isn't just my seed treatments. This is herbicides, foliar fungicides, all sorts of things. So again, there's corporate decisions on working with some of the crops, and in particular the hemp is another hurdle, is just working with the corporations, and they're willing to support labeling that they can be used commercially. Again, in this particular studies, none of the biologicals were giving good control. We're moving to yet another case study, and this is on the use of biostimulants. So biostimulants is a broad class of materials that are really used to enhance plant growth. In this case, we're looking at germination or seedling growth. We're kind of revving up the plant to kind of enhance the vigor, so to speak, of the seed. There are many different categories for these. The microbial, and again, we heard that touched upon in the earlier talk, humic acid. One that we've kind of worked on are more of these protein hydrolysate materials in coatings, seaweed extracts. So again, we're not controlling insects and diseases now. We're really trying to do something to benefit the physiology of the germinating seed. Where most of the biostimulant research is being done at is on foliar applications. So that is where we see the primary use of the technology is used as a foliar. Some with soil application, but much less on seed treatments. But we really consider that as a big opportunity as we move forward. So here's some of what the treatments look like. And again, a lot of acronyms, so I'll just go through this at the bottom. The SF stands for the soy flour. So what we're after here is we're looking at the protein from the soy flour. These are defatted soy flour. Also, the soy flour acts as the binder, so it's a dry binder, so we can develop our coating with this using the soy flour both as a biostimulant, but also as a binder. The DE, diatomaceous earth, is commonly used. And then some of the materials that we've used, this MVC, is a vermicompost from different sources. So again, a lot that went into this, and we published just a few years ago. But to look at the potential with the same variety, and this is all broccoli seed for these comparative studies. I know there's a lot on here, but kind of look at the root and the shoot growth compared to the control on your left. The 2.7 is a 2.7 centimeters height for the hypocotyls, 9.3 centimeters for the roots, but look at the enhanced root and shoot growth from these various combinations that we're testing. This is just 10 days after we do a germination test where we've taken the seedlings and then just ranked them from high to low. So a number of those, even in the control, that the very small you would consider as abnormal seedlings, now because of their increased seedling length, you would no longer call them an abnormal in a standard germination test. Taking this into the greenhouse for transplant production, here we're looking at just growing plants individually. So we're seeing improved shoot growth, root growth, leaf area. So we're seeing improvements in several different parameters, photosynthetic measurements and things like that. So quite an enhancement that we're seeing here from our biostimulant seed coatings. And then removing those plants from the pots, we see the enhanced shoot growth, but also the very enhanced root growth. So this we're really excited about this potential of what we can do here as a biostimulant. Now, as I've given talks to people in my department in horticulture, they say, well, you're applying nitrogen, you know, you have proteins and things. You're really just putting a nitrogen fertilizer on these. And I said, okay. So we're going to test that. So here we're looking at kind of like a soil, kind of a plant nutrient analysis of the vermicompost or the soy flowers. And yes, we do see a lot of protein in the soy flour, which you would expect in some of the different constituents. But our research question is do the seed coatings serve as a nitrogen fertilizer, or are they really acting as the biostimulant? Now, if you look at this, we can see the two treatments, the soy flour DE and the soy flour, the MVC. The nitrogen applied per coated seed is 0.05 milligrams for the soy flour treatment. With the vermicompost, we're up to about 1/10 of a milligram. But now from our greenhouse grow out, the enhanced nitrogen uptake is at 1.5 to 2 milligrams. So we can only, you can only account for less than 5% of the nitrogen from the coated material itself. So we're looking at truly as a biostimulant effect going on here with the treatments. So just to summarize here, the development of the new and effective delivery system with the biostimulants. We feel that the soy flour, the vermicompost, really for small seeded, high value vegetable crop seeds could be used. Also, I did not show the data, but we had really a negligible effect on the germination parameters. It did not slow down the germination, did not affect the T50 values, did not reduce germination. And that was a big accomplishment by the formulations that were developed in this. So we see the co-application of the two biostimulants, both the soy flour and the vermicompost, kind of give you a kind of a double whammy effect of improving the germination. Okay, we're in the last case study here. And in the case of growing cover crops, and cover crops in the United States is where you're growing the main crop, such as corn, and but as the corn grows, where you at the time about where you're going to side dress, then you'd want to go ahead and put the cover crop seeds in between the rows to help stabilize soil, do other improvements that the cover crop can do. The thing is if you're going to broadcast those cover crops, they're going to be on the surface. The soil can be dry at this time. So our interest was to add a hydrophilic polymer or a superabsorbent polymer, you'll see SAP, to absorb soil moisture to help overcome the deleterious effect of a dry soil effect on germination and stand establishment. This has been used already on lawn seed. They can buy these in the United States with already seeds are already, the lawn seeds are already treated with a hydrophilic polymer. So he says, well, it's already been done in that, we can do this. So we kind of adapted that technology in our laboratory for small scale application using red clover as our model system. This is what one of the polymers look like. These are the same type of materials that are used, say, in baby diapers and things like that. They're hydrophilic polymers. They absorb, over 200 times their weight in water. So they have a very high water absorbency. A number of materials that we've used, and trade names, the stock absorb, and the trade names, but it's a cross-linked polyacrylate. We're looking at polyacrylamides. We have starch-based polymers, and one is a polyacrylamide with a graphite that's added in with the coating that's commercially added in. So I mentioned about this water absorbency. Working with these companies, we kind of adapted their volumetric liquid absorption, so we have a protocol so we can go ahead and take the polymer, let it take up water, and then it's going through this mesh screen to see how much water is being retained by the polymer. So we have a way to actually compare one polymer against the next. Let's look at some of the data here with the polyacrylamides or the polyacrylate materials, and then the starch material having the highest. So again, we're looking at over 200 times the water absorbency, over 200 times their weight taken up in water, over 350 times with the starch-based polymers. Now to test these in the laboratory, we're using kind of a soil media, which is montmorillonite clay particles, which have high water, but they would also freely give up the water, but not cause a hard crusting. They'll still remain friable. So we hydrate these, put the coated seeds on, then dry them, and then kind of seal. So this is an enclosed type of a system. So we have a very controlled environment of a wetting and drying. The reason we do that, these polymers are not taking moisture up out of the air. They're not hygroscopic. They're hydrophilic. There has to be free moisture there for it to be taken up. So that's why we're starting off with this wet soil and then drying the soil to produce a drought situation. We can look here with the different treatments, and something got a little bit crazy with this slide, but just know that the black and the purple are the control or just the coated only. All those other colors are some of the different polymers up above, which have the hydrophilic polymer in there. So indeed it was working under our controlled lab environment situation. There's a lot of text here, but let's just kind of look at the pictures. So we had 3 plantings, a very dry soil situation, planted in June of last year, a kind of intermediate soils with moisture coming in, rains coming in, and then September when we get more of our rainy season in the moist soil. So we kind of have naturally 3 different soil conditions dry, kind of an intermediate, and a moist soil. We're just kind of pulling treatments out, some of our better treatments. The trial one, very dry. So you see the very low percent stand. So what we're seeing, and then we also had either broadcasts, which is a capital B, or the BR is the broadcast, then raked in. So we're basically, with the addition of the superabsorbent polymer, we're able to make some improvement in sand establishment of the cover crop. We were not able to overcome the complete drought stress, but we were able to make improvements. And I think that's kind of, we're looking at this as kind of as one tool to help overcome drought stress, but it's not going to overcome all of the drought stress potential. So really what the moisture needs to be there to begin with in nature, and we can help that, but we can't eliminate that water stress. So kind of just the summary, we've developed an effective coating system with the superabsorbents. We feel like in certain situations where there's a kind of a transient type of a short-term type of a drought stress, it can help in those particular situations to improve the stand establishment in those. And just really thank some of the people in my lab, plus just a number of other people and the funding agencies involved behind us with this. So I just want to open it up into questions, and I need to look at, I see a few things in chat. I need to look at that at this time if I can respond to those. Thank you for your attention.
Thank you very much, Professor Taylor. Is there any question? Yes, Dr. Gülay Beşirli. Dear Dr. Taylor, thank you very much for your interesting presentation. It is very exciting to see and hear you after side of the world. I am Dr. Gülay Beşirli from Turkey and an onion breeder. We have onion maggot problem for keeping our very valuable meal lines in the field. Then after harvest chipping of the bulbs, I want to ask the processing of the spinoza using.
Okay, so through our research, where we started working with our vegetable entomologist, we did all the efficacy testing, and it was tested in other states, even in Canada, the country just north of us, have similar pest problems, including onion maggot. And the spinosad is a very effective material for that. I think what would be good is I provided my email address. We can have some email after the conference sometime, and we can talk to more about the rate of applications and things like that. But I think you could find that, for onion maggot, Delia antiqua, it's a very, very, very good and very safe material to work with as well. These are organically approved materials as well.
Okay. Thank you very much. Any question? Any other question? Dr. Beşirli is writing.
Okay, thank you.
One question, but I think. No, he is thanking you. Yes, Professor, you look great, really, and it is honor and nice to see you and listen to your presentation. Thank you again.
Okay.
Okay, the next speaker, Professor Philip Simon. I think he is not ready at this time. Abi yok herhalde.
Hayır hocam, sistemde görünmüyor. İsterseniz bir sonraki araştırıcı geçelim.
Okay. The next speaker, Dr. Stephen Putit. Dr. Putit is currently working at Department of Molecular Biology and Genetics, Boğaziçi University, Turkey. He has done his bachelor degree in field of applied biology at Northeast London Polytechnic, London, UK. He completed his PhD at Louisiana State University, Louisiana, USA. In addition to that, his recent interest on change in phonological events in response to a global warming scenario. He mainly works on seed dormancy and dormancy cycling in the relevant field. He has published many papers in reputed journals. Now his presentation topic is physiological seed dormancy in a changing climate. Yes, Dr. Steven, please.
Okay, can you all hear me?
Yes, yes.
Okay. Okay, I'd like to thank the organisers for giving me the chance to speak to you all today. And as it's just said, I'll be talking about physiological seed dormancy in a changing climate. So as I'm sure you're all aware, climate change poses a serious threat, not just to our native flora, but to also agricultural production. And what I'm going to talk about today is some of the effects we've seen of climate and temperature, to be specific, on dormancy, and how we can potentially exploit this by looking for climate adapted genes in wild species. In my case, this is Arabidopsis, and these have potential for helping us to identify genes which could be important in breeding programs, so we can produce the climate smart crops that we're going to need in the next few years. So what's physiological dormancy? So physiological dormancy is an innate property of the seed that blocks the capacity to germinate over a specified period of time under any combination of environmental conditions, for example, water, temperature, oxygen and light, that would normally support the germination process. So within this, we have two types of physiological dormancy. So we have primary dormancy, and this is imposed on seeds during their maturation program. And the depth of this dormancy is determined by the interaction of genes and the environment during seed production and maturation. When these primary dormant seeds are then dispersed from the mother plant, they can quite quickly lose primary dormancy. And if adverse conditions prevent germination, dormancy levels then increase and seeds start to undergo dormancy cycling. And this is what we call secondary dormancy. And this secondary dormancy can be far deeper than primary dormancy and far more persistent. And this is what leads to the accumulation of highly persistent seed banks in the soil. Oops. So when seeds enter secondary dormancy, they cycle through periods of deep and shallow dormancy, and this is dependent on the season of the year. When they're in deep dormancy, seeds become insensitive to environmental signals that would normally break dormancy and enable germination, but they are sensitive to temporal signals such as temperature that indicates the time of year, and this is what results in the increase and decrease in dormancy during cycling. As dormancy decreases, we enter a shallow dormancy phase. And here dormancy again is declining in response to the environment, and seeds become spatially sensitive to environmental signals that indicate they are in a suitable position for germination and seedling growth. Now these spatial signals, they tend to operate in a particular order. So seeds become sensitive first to temperature signals, then nitrate signals, and then light. And these indicate for example, the depth within the soil and the presence of other plants. And these signals ultimately remove the final layers of dormancy to allow the initiation of the germination program. So for the past few years, myself and my colleague, Bill Finch-Savage, we were using natural variation to understand the dormancy regulation. So in particular, we used natural variation between populations, and this helps us to understand the adaptation to climate. So in particular, we used winter and summer annual Arabidopsis ecotypes to show the seasonal organization of global gene expression during seed dormancy cycling in field soils. So if we have a quick look at these winter and summer annual ecotypes, so in these, their life cycles are adapted to diverse climates. On the left here, we have an example of a winter annual life cycle, and this is from an ecotype that originated in the Cape Verde Islands. So in this winter annual, we get out in the field, this is particularly in the United Kingdom, we get seedling emergence in autumn, and then overwinters as a rosette before flowering in the spring, and then dispersing its seeds to the soil in late spring and early summer. Now we have the impact of warm soil temperatures. So now the seeds are in a shallow dormancy phase, and they are able to sense spatial signals, and ultimately they will then lose dormancy and germinate, and we get seedling emergence in the autumn again. So if the environmental conditions are adverse to germination, then the seeds go more dormant, and they enter the deep dormancy phase in the persistent seed bank, and they go round and round in this temperature-driven cycle until they either germinate or die. So if we then look at the summer annual life cycle, this is essentially reversed. So this is in the Burren ecotype, which exhibits this type of life cycle. In this ecotype, now we see seedling emergence in the field in sort of late spring, early summer. And then the flowering is in the summer, and the seeds are dispersed into the soil in autumn. Then the seeds go into a sort of like deeply dormant, but then quickly into a shallow dormancy phase where they can sense spatial signals. But then if the soil conditions are adverse to germination, then these seeds will also go into a deep dormancy phase, and they'll be then in the persistent seed bank. And then they will also go round and round in this temperature-driven cycle until they either emerge or they die if they just run out of energy. So how do they come by these adaptations? So the Burren ecotype, this comes from the Burren region in the west coast of Ireland, and here the climate is cool and damp, whereas the Cape Verde Islands are off the west coast of Africa, and here the climate is hot and dry. So we took seeds from these two ecotypes, and we produced them in a glasshouse in England, and then we dispersed them out into a field plot in the field trial area at Warwick University. And so this was in November 2009. And what we found was that in the Burren ecotype, emergence was in May and June. Seedling emergence occurred in May and June, and this was typical of an obligate summer annual life cycle. Whereas CVI, here the seeds emerged in August, early September, and this was typical of an obligate winter annual life cycle. So these annual behaviours in CVI and Burren that we saw in a common field is an adaptive response to dormancy cycling to climates separated by 37 degrees of latitude. So we were then interested in how do these climate adaptive ecotypes respond to temperature. So first of all, we were interested in the impact of temperature on primary dormancy, in particular its response to global warming. So if you remember, primary dormancy is induced during seed maturation. So we took these plants and we grew them up in a thermogradient tunnel that was available to us at Warwick University. So here at this end we have the ambient temperatures. It's just the large tunnel you would find in horticulture. So it's open to the atmosphere. At this end, it's just an insect screen to protect it. Plus at this end, we have heaters and fans and sensors down the middle. And using this equipment, we could establish a thermal gradient from ambient at the cool end to +4 degrees at the warm end. So this is the cool end, the warm end, and this is in the winter. December, and this was in August, in the late summer. So we were able to maintain this thermal gradient throughout the year. It just moves up and down in response to the ambient temperature outside. So we ran this experiment in 2011, and so the thermal gradient of 4 degrees represents the impact, the predicted impact of climate change if we don't stop emitting greenhouse gases between 2011 and 2080. So we grew these plants at 4 positions along this tunnel, and we did 2 different sowings. So early sowing was in April, and the later sowing was the end of May, beginning of June. And what we also measured, because we measured recorded air temperature, we were able to calculate the mean temperature during seed maturation. And we found that when we looked at germination, when we harvested seeds, we tested germination in the light at 10 degrees and in the light at 25 degrees. So we found an overlap in the maturation temperatures in the 2 sowings, which was very useful. When we looked at germination at 10 degrees, seeds produced at the lower mean temperature here, they germinated about 50%, and they germinated around about 90% at the warmer temperature. The warmer range of maturation temperature. But when we looked at seeds germinated at 25 degrees, we saw that the seeds which had been matured at lower temperatures, these now had demonstrated high temperature thermodormancy at 25 degrees, but this was lost in seeds which were matured at the higher range of temperatures. We also repeated this on the more dormant CVI ecotype. So here we had to, because these seeds are so dormant, we wouldn't see any germination straight off the plant. We have to ripen these for 30 days. And then we incubated them in the light at 15 degrees. And again, we saw relatively high lower germination when seeds were matured at a lower temperature. But then as the maturation temperature increased, dormancy levels were lower. So we saw dramatic nonlinear effects of maturation temperature on dormancy. And what was even more interesting, that we observed a thermal switch in dormancy, which seems to occur between 15 and 16 degrees centigrade. So there's a switch occurs here, and we can see it's here between 15 and 16, and also in the CVI ecotype. There's a switch point here. So this was quite interesting, and it became even more interesting when we saw data from colleagues of ours, the Steve Penfield's research group at John Innes Centre. At the same time, they were doing work looking at the impact of temperature on flowering time in Arabidopsis, and they grew plants at 2 different places in England, measured the temperature as they recorded flowering and the time to first seed set. And when they modeled this against the thermal gradient going from north to south in Europe, they found that the temperature induced flowering occurred so that seed development, seed set, would occur at a mean temperature of about 15 degrees. And this was a bet hedging strategy, so that when seeds were dispersed, part of the population would be highly dormant and part of the population would have low dormancy. So this was essentially what we were seeing when we matured seeds at different temperatures, this response. So what happens when to primary dormancy at the temperatures we find in Turkey? So here we have to switch species, and now we're looking at Amaranthus retroflexus. This is from species is from Central and North America, and it arrived in Turkey in the late 1800s. So it's considered the most economically important weed in Turkey, if not the whole world, and it causes large reductions in crop yield, as we can see here in this tomato field in Eskişehir, in central Turkey. So Amaranthus is a C4 plant. This means it's adapted to high temperature and drought, and its seeds are also very dormant and can survive for up to 40 years. So how does primary dormancy in this species respond to temperature? So this data is provided by the Ultrafootic Research Group at Dunlupinor University. For several years now, they've been looking at adaptation of Amaranthus within Turkey. So in this experiment, they collected seeds from nine cities, mainly from agricultural fields on the outskirts. For example, Afyon, Bursa, Eskişehir, and as far east as Mersin. So they germinated these seeds in the light and dark at 25 degrees centigrade in the presence of 100 micromolar gibberellic acid. And there's some positive effect of light, but not a great deal. But the interesting thing was that when they plotted germination against the mean temperature over 60 days prior to harvest, what they found was that germination was lowest at the lowest mean temperature of about 17.5 degrees centigrade, but as the maturation temperature of these seeds increased, primary dormancy got deeper and deeper, and so it reached the deepest point here, and this is in seeds from Bursa. But after this point, it seems that another temperature switch may well occur, as the seeds from Mersin, which were matured at the highest temperature, these now had a decrease in primary dormancy. So this data indicates that with climate change, as the climate warms, amaranth may become even more of a problem, as seeds will become even deeper primary dormancy when they're shared into the soil, and this means that they may well this may contribute to increasing the size of the long-term seed bank. So when I was in England, we were also interested in looking at how secondary dormancy responds to temperature in these climate-adapted ecotypes. So at this time, little was known about how secondary dormancy operated in the natural environment. So we buried CVI and burren seeds out in the field, and we recovered them every month. And this was done we developed a way of digging the seeds up in the dark, so light didn't affect the dormancy level. And then we did physiological experiments on them, and we also wanted to ask what for us was the big question is, how does gene expression differ in buried seeds of ecotypes which are programmed to emerge at different times of year? So if we have a look at some of this dormancy cycling data. So first of all, this is for CVI. So here we have this is the time that the seeds were buried. So here we buried them in October, and this experiment lasted for 12 months. The red line here, this is soil temperature, shown here. And this shows the low temperature in the winter and as it gets warm in the summer. We also measured the level of dormancy in the CVI ecotype, which is very dormant. So these red symbols represent the level of dormancy. And this is shown here. And this was determined by measuring the after ripening time to give us 50% germination. So when seeds were first buried, it took about 50 days of after ripening to give us 50% germination, but after 2 months of burial it took over 150 days. But then this dormancy then started to rapidly decline between April and May, and by July and August there was no need to after ripen seeds, as just exposure to light would break dormancy and cause them to germinate. Then looked one of the main genes we wanted to look at was DOG1. So this is the delay of germination 1, and this has the strongest association with dormancy based on QTL analysis. And this was done by Benson et al. During her PhD. And they also cloned it, and they show that the mutants have very low levels of dormancy. So when we looked at this gene, very low levels here in primary dormant seeds, but when they entered secondary dormancy, the levels increased, and they follow the opposite pattern to soil temperature. So it appears that DOG1 is correlated negatively to soil temperature and positively to dormancy, because it tracks the same pattern of dormancy in these seeds. And so in the deeply dormant CVI ecotype, dormancy and DOG1 appear to be driven by temperature. Then we looked at the Burren ecotype. So again, this experiment ran from October to October of the next year. And again, this is the red line is the soil temperature, showing winter temperatures and summer temperatures in the soil. We also looked at the depth of dormancy in Burren. So here Burren is only weakly dormant, so we can't really do a dry ripening after ripening test on these seeds. So we had to measure base water potential. So the more, so this is the scale, sort of in megapascals. So the more positive the base water potential, the more dormant these seeds are. So here we see dormancy is increasing as soil temperature is low, and then as it starts to increase, we see a decline in dormancy. But this does not seem to do the same pattern as soil temperature. And we also looked at DOG1 again. This is the black symbols, and DOG1 again, it follows the opposite pattern to soil temperature. So DOG1 expression correlates with negatively with soil temperature, but it doesn't correlate with dormancy at all. So it appears that in DOG1, in Burren, DOG1 and temperature don't explain the pattern of dormancy. So to understand this, we looked at compared full genome expression patterns using RNA-seq during these annual dormancy cycles. We then used cluster analysis to identify seasonal timing of gene expression. But before we look at the RNA-seq data, we should say something briefly about other processes which are important in this process. So Gerd Lubner talked briefly about the hormone balance theory, which he and Bill Finch-Savage put forward in 2006. So basically, in response to the ambient environment, seeds either go more dormant or they lose dormancy. And as they go more dormant, this is involved in increases in ABA synthesis and gibberellic acid degradation. And we get increases in ABA sensitivity to ABA and increased signaling. And this results in seeds becoming more dormant. And of course, DOG1 is very intimately involved in this process of increasing the dormancy of seeds. But as they start to lose dormancy, we get increased GA synthesis, ABA degradation, and increasing sensitivity to GA as the GA signaling pathways become active. And so as a result of this, we have this sliding pattern of changes of sensitivity during annual dormancy cycling, and this alters the sensitivity to the germination environment. So when seeds are deeply dormant, they have little or no sensitivity whatsoever to a germination environment, but this gradually increases as dormancy decreases, as this balance changes. So we go from perception to integration and then to the response during cycling. And in this whole process, we have hormone signaling. We have large numbers of genes that contribute to the regulation of these opposing signaling pathways. And this is what we started to look at when we had the RNA-seq data. So when we analysed this data, we found 9 clusters. And to understand the significance of each cluster, we identified transcripts with known roles in the maintenance of dormancy and the germination process. So here we have the clusters listed for Burren and CVI. So they're different. So this is these shaded areas represent autumn, winter, spring, and summer, and these dashed lines represent soil moisture. This is increasing cold, and this is increasing warmth. So this is summer, and this shows winter here. And the green dashed line shows when we saw increasing germination, when we did germination tests, and the red arrow shows when we saw seedling emergence out in the field. So what we find is these major gene clusters are segregated either with environmental variables or with ecotypes. So clusters 1 to 4 are associated with winter. And each cluster contains genes associated with dormancy and ABA responses. So NCED6 is a key gene in ABA biosynthesis. DOG1, key gene on the regulation of dormancy. MicroRNA 156, this blocks phase transitions, so it blocks the expression of genes that will be related to germination, for example. ABI1, a phosphatase, which downregulates ABA signaling. And here a gene which is involved in ABA degradation. So these first two clusters here, cluster 1, this is associated with changes in soil moisture, and it occurs in both Burren and CVI. The second cluster, again, this is driven by increasing cold. So this is in winter in both ecotypes. But then we have this cluster 3, which is only associated in the Burren ecotype. And then another one here, cluster 4, this only occurs in the CVI ecotype. So these two clusters here, cluster 3 and cluster 4, these indicate climate adaptation in the two different ecotypes. Right. Okay, so this slide's changed. Okay, so cluster 8 is associated with warmth of summer. So this is the wrong image here. Just let me go back. So here, cluster 8. You can see it down here. So this is a cluster which occurs in both Burren and CVI, and this is an increase in cohort of genes whose expression increases and this is related to the heat of summer. So this cluster contains genes which are related to shallow dormancy, and this involves DELA protein. The genes encode DELA protein. So DELA proteins, they repress gibberellic acid signaling. So this indicates that if these are increasing, this indicates that seeds are poised to germinate, and they're just waiting for the final environmental signal to activate the GA receptor, which will then bind to these proteins and causing their degradation and release the suppression on GA signaling within the seed. So then we get to the 4 clusters which are associated with ecotypes. So here in cluster 5, again, this is so I'm sorry about this. The slides have been altered since I sent them. Okay, so cluster 5, which is this one. Cluster 5 is down-regulated, so here in this point, it's down-regulated when dormancy increases in Burren, and it increases when dormancy increases in CVI. Then this contains genes related to associated with ABA, jasmonate, and ethylene responses, and it's related to changes in dormancy and operations within these ecotypes. In cluster 6, here genes are positively correlated with shallow dormancy. So this is shallow dormancy here, and here. And these genes increase, their expression increases as we see increases in germination potential when we test germination in the laboratory, but also they're associated with the point where we start to see seedling emergence out in the field. And then cluster 7, here the genes are associated with seed maturation. So this is cluster 7. So this is associated with seed maturation type genes, and these include storage proteins and heat shock transcription factors, and their expression peaks prior to or just before we see the loss in dormancy. So if you look at this, the green line here, this shows where we will see germination in the laboratory when we break dormancy. So this cluster here is increasing associated with this change in physiological response, and also here in CVI. Then cluster 9, here the genes are strongly expressed in germinating seeds. So, well, this is when seeds will germinate. So we have to remember that at all times the seeds in this experiment were always dormant. So this cluster here is genes which are associated with germination. So here their genes have been expressed. So this is at a point where seeds are becoming poised, so that all they need is the final spatial environmental signal, the spatial signal, which removes the final layer of dormancy and allows them to germinate. For example, when the soil is disturbed and they see light. So then potentially then these genes may not be been transcribed. So this may be been transcribed but not translated, for example. So these are they're waiting poised to allow for when the germination program is initiated. And we only see this cluster in burn seeds before they're buried. And this indicates the differences in germination potential that we see between these ecotypes. So what do these gene clusters tell us about climate adaptation? Well, they tell us that the environmental signals, such as temperature, drive the expression of specific clusters, and these are common to both ecotypes, both Burren and CVI have these climate-driven clusters. And this represents the monitoring of the seasonal environment. So these are common features which make seeds highly adapted environmental sensors. The remaining clusters are ecotype specific, and they determine how the seeds respond to the environment. So this gives us an indication of how the responses have been adapted by climate. And finally, climate adaptation in these two ecotypes has driven temporal separation of gene expression. So are these ecotype associated clusters important? So they indicate that climate adaptation of obligate winter and summer annual ecotypes has altered their life cycles, their phenology out in the field. Another important question is, because these are wild species and we're looking at natural variation, is this relevant to agriculture? Well, we would say yes, it is, because this work may help identify genes that have the reduced potential for secondary dormancy induction, or to extend the thermal range of germination. And in crop plants, as in unfavorable climates, secondary dormancy is a serious problem in things like oilseed rape and sunflower seeds, sunflowers, and also when the thermal range for germination is reduced, we get low vigor, and by identifying genes which can extend this thermal range, we can increase the vigor of our crops. And as is quite widely known, increased vigor is one of the biggest determinants of maximizing yield. So altogether, these genes, climate adapted genes, which are present which we hope are present in these ecotype related clusters, may contribute to increased seed vigor as we try and develop the climate smart crops that we're going to need in the next few years. So to address this, so I've been in Boğaziçi now for 2 years, so I arrived only marginally before the COVID did. So for the last 2 years, we've not been able to do any work, but we've just started 2 projects. So the first project is to identify QTLs that regulate temperature-sensitive induction of secondary dormancy, and this is using an Arabidopsis mapping population, and this is funded by the BAP Commission at Boğaziçi. And in the second project, we'll be trying to identify climate-adaptive genes that regulate induction and relief of secondary dormancy in these Burren and CVI ecotypes. And this will be focusing on identifying climate-adapted genes that are expressed separately in the embryo and the micropilot endosperm of these seeds. So finally, I'll thank you for listening to us. At the moment, we're only a group of 2, myself and Ataline Nalader, but hopefully in the not too distant future, we'll have a postdoc working with us. And these projects will be, we have collaborators in the UK, Dr. Peter Wally, and who's doing QTL analysis, and a bioinformatician, Dr. Stuart Lucas at Sabancı University. And I'd like to thank our funders, BAP and TÜBİTAK. Thank you very much, and if you have any questions, I'll be happy to try and answer them.
We thank you very much, Dr. Steven. I see Gerard Lebner, Dr. Lebner's writing something.
I may better ask it in person because I'm typing so slowly. Nice to see you, Stephen.
Yeah, nice to see you as well. Good.
Great to see you. Yeah, I very much like this research about the cycling. How easy is transferring genes which one finds in Arabidopsis to weeds and crops for the same purpose? How, if you find something in the summer and winter and your Arabidopsis, how easy is it to say about a weed or a wild species that these genes are involved in this?
Well, because we have these specific gene clusters, that helps us to narrow down where we're going to look in the genome. But with Arabidopsis, it's also a member of the Brassicaceae, so any crops within the Brassicaceae, we should expect to find similar genes within them, although it would be a bit more difficult because the ploidy level is different in many of the crop Brassicaceae. For example, oilseed rape and Brassica oleracea. You have they're like tetraploid and hexaploid, some of them. So that makes it a bit more difficult to pin down the actual gene which was present in their multiple genomes. But it is possible, and it has been done before, going from models to crops. Bill French Savage, they did it in a project, but it took them a long time. It took them 9 years to go from identifying a gene which increased vigor in Arabidopsis to identifying the same gene in a Brassica mapping population. But it is possible. I mean, it is difficult. It's not straightforward.
Okay, thank you.
Okay. Any question? I think no. Thanks again, Dr. Steven, for your presentation.
Okay, it's my pleasure.
Our pleasure. Thank you. Yes, the next speaker, Professor Philip W. Simon. Dr. Simon, I know him for sending his carrot genetic resources to our country, and I thank him for contributing carrot breeding in our country. He is a professor of horticulture University of Wisconsin, Madison. He has obtained a doctoral degree at University of Wisconsin. He has been working as USDA Research Genetics and Professor of Horticulture, University of Wisconsin, Madison. His main researches are addressed on development of genetic and genomic tools. He has also contributed in genetic improvement of market carrot germplasm. Yes, his presentation topic is putting the carrot genome to work, developing better tools to build better carrots. Yes, doctor. Go ahead, please.
Oh, can you hear me?
Yes, we can.
Very good. I can see my presentation, so thank you very much for setting this up, and also thank you very much for the invitation to speak at this meeting. It's an honor to so, and it's especially an honor given the strong history of carrots in Turkey. I'll come back to that as I speak about our work, but what I'm going to talk about today is what we've been doing since the carrot genome has been sequenced and how we're using that sequence to move forward in improving the crop. So with no further ado, let me see if I can change. I'm not sure if I can change slides. Maybe this can do it. Yep, there we go. So of course, the genome is this complete set of genes that occur in the chromosomes of an organism. In the case of carrot, there are 9 chromosomes, and we're doing a lot of work to evaluate the genomic sequence on those chromosomes. We've started this work quite a while ago, and as I mentioned earlier, the sequence was completed in 2016. So here we see some chromosomes as they appear in a carrot cell, and also the circles to the right represent some carrot chromosomes with the individual chromosomes around the edge of the circle. The carrot genome sequence was based on a doubled haploid that Reich Juan provided to the project, and this gave us complete homozygosity for the plant that we were sequencing. In this process, we discovered over 30,000 genes, including some extensive repetitive sequences of all other genomes. We accurately assessed the size of the genome, and we identified in this particular project the function of a gene controlling, one of the major genes controlling carotenoid accumulation, those orange pigments that occur in carrots. So with this project, let's see, get the next slide going. There we go. Since this project, there have been roughly 300 publications citing the work of 2016 on topics regarding carrot pigments, flavor compounds, terpenoids, some new work on genome editing, looking at genome structure, transposable elements, root development, taxonomy of the crop, but also this information is applicable to other members of the APAC, of which carrot is a member, other crops like celery or coriander, but also to other plants as well. So where have things been moving since then? Well, when we sequenced the genome, in addition to the sequence of the doubled haploid, which is the outer reddish ring on this circle, we also resequenced 8 wild carrots and 18 cultivar land races of carrots, inbreds. And in this process, not surprisingly, we discovered a lot of genetic variation. And these inner circles on this diagram indicate that variation of those 18, or actually 26 other carrots that we had resequenced relative to the sequence of the doubled haploid around the edge. So this variation that we observed initially, knew about before from just doing genetics of carrots, this variation is what we're interested in harnessing to improve carrots, to make it a better crop than what it is today. Let me give a brief overview of where we are at in terms of the genetics of carrots. Back in 1986, I was involved in a publication where there were 21 traits noted to have a genetic control of them, but no genetic markers were known at that point yet in 1986. The first genetic markers were actually isozymes, 1991, about a dozen of them. The first traits that were looked at were in the early 90s. And you can see with this list of genes and traits here, that as time has gone along, carrots started out very slow about 40 years ago, but we have accelerated things quite quickly with sequencing the genome, so that today we have about 60 traits that we have some genetic mapping for, and we're using genotyping by sequencing to generate more information rapidly as we go along. These mapped genes that we're discovering are of course, what we're using to facilitate the breeding and selection of carrots. So let's take a look at the history of carrots. Carrots first are regarded to have been first domesticated as a root crop and grown ever since for about 1,100 years, so not near as long as many crops. And in the process of domestication, this wild carrot that's on the left photograph here has been improved to a range of different cultivated carrots, many of which are orange, but there are, of course, purple carrots, which are widely grown, I know, in Turkey, but also yellow, red, and white carrots. And so as carrots were domesticated, some of this genetic variation was harnessed to be incorporated into the cultivated crop that we see depicted with this ring of carrots to the right in this circle, to the right. And so there is a large amount of genetic variation in carrots. This is what's been used, starting with this wild carrot, often called Queen Anne's lace, that I showed you in the last slide. Starting with that crop, somebody 1,100 years ago was brave enough to start eating that crop, probably in Central Asia, places like Uzbekistan, Afghanistan, and the crop was domesticated in the process. And as I mentioned before, Turkey played a major role in the domestication of carrots. Lots of historical records for carrots in Turkey. But the question that comes up with carrot improvement is, who needs more carrots? You go to the grocery store, there's carrots there every time, I'm sure, in Turkey as well. And what role can carrots play in feeding a growing global population, and what future production challenges might be faced by carrot growers and needed by carrots consumers? Let's take a look at carrot production in the last 50 years based on the global statistics available. There's more information here than I want to talk about, but the important things to look at are on the column, where globally the production area of carrots has increased about threefold in the last 50 years. At that same time, the yield per unit land area has roughly doubled, so that globally the carrot production is about sixfold today compared to what it was 50 years ago. And so this translates to a 2.7 or 2 to 3-fold increase of carrot availability per capita to the global carrot consuming community, assuming everyone consumes carrots equally. Next slide here. So if we look then at the rise of carrot production, the rise was relatively slow in 50 years, but in recent years has climbed significantly. Let's take a look at where carrot production has increased most significantly globally. As I showed in the last slide, 2.5 to 3-fold increase of carrots per person around the world in the last 50 years. Not much of a change in that 50 years in Africa and the Americas, in Europe and Oceania, but the big change has been in Asia. So that's where the big boom in carrot production has occurred in the last 50 years. And this is an important factor when we think about global health that I'll talk about in the future here. So what does a carrot crop look like today? Typically, if we go around the world, in Asia, the typical carrot shape, first of all, carrots are orange, but the typical shape of orange carrots in Asia is typically this Carota type. In Europe, it's more often the Nantes type. And in the US, it's the long, thin Imperator type. And so there are different types around the world, but there are carrots adapted for all these shapes, and for that matter, different colors all around the world. And as time has moved along, there's more of a trend towards the consumption of fresh or raw carrots. So the demand for carrots has increased in the last 50 years, and we can expect that increase to demand because there will be more increases in populations. And the question is, how can we use this genetic variation that we started looking at very seriously with the sequencing of the genome? And we're looking at genomic applications to improve the crop for both consumers and, of course, to improve the crop growers. Let me talk about improvements that there's been some work to initiate improvements on and where we're at on those, and then talk about those for growers as well. So looking at improvements of carrots for consumer quality, from the U.S. perspective, U.S. consumers are interested in convenience as a major characteristic of carrots that's been improved in the last primarily 25 years. So a big part of the U.S. carrot consumption today is baby carrots, which are shown in this photo to the right, are these short pieces of carrots. These are not whole carrots, but long carrots cut into pieces and peeled slightly. And this has been a major convenience product for U.S. consumers. If you've been to the U.S., you're aware of the fact that they're more expensive than the same cost per unit weight of whole carrots, but people are willing to pay for them for the convenience. So convenience has been one thing that U.S. consumers have been interested in when we look at carrots. In the U.S., 85% of the carrots grown are for fresh market carrots, or they're carrots that are sold as either whole carrots or as raw carrots in pieces, like we're looking at here. So carrot consumers, in addition to that convenience, not surprising, are looking for better flavor and texture. We've got some significant genetic evaluation and significant genetic variation for flavor and for texture of carrots. The figure to the upper right shows you some variation in carrot sugar breeding over 5 generations of selection. Carrot consumers are always interested in sweeter carrots, and that's one of the traits that we're looking at for improving consumer quality. We're also looking for uniform orange color from a consumer stand. That's important. And the consumer, in addition to wanting this uniform color, this color of carrots is a significant nutrient for U.S. consumers, and for that matter, for anyone who eats carrots around the world. This is important. The nutrient of particular importance is vitamin A, which is a derivative of the orange pigments that we eat in vegetables like carrot or sweet potatoes, or in crops like orange melons. So there's a significant need for vitamin A in all diets. There's a borderline need for U.S. diets, and there's a significant need in much of the developing world because of its importance in prevention of blindness and also providing immunity to certain diseases. So we're very interested in tracking the nutritional quality of carrots as a consumer trait, in addition to those traits that consumers can overtly evaluate, like flavor and texture and convenience. As we look at this, we're looking at the question of what can we do to enhance carrot consumption by putting carrots in different colors that might be of interest to consumers, and I show you some of those colors to the right. But from a nutritional standpoint, it's the vitamin A that can be derived from orange carrots as of particular interest. If we look at what the role carrots play in providing pigments, let's take a quick look at the colors of carrots. Carrots, as I mentioned, were domesticated as a root crop roughly about 1,100 years ago. Made to be probably in Afghanistan, spread to the east and the west from there, spent a long time and a lot of development in Turkey. And along the way, these different colors of carrots, yellow with lutein, red carrots with lycopene, orange with alpha and beta carotene, and purple carrots, which in addition to those carotenoids can include anthocyanins. So orange carrots first showed up only about 500 years ago in Southern Europe, and have been important for the development of carrots globally, because as I mentioned before, most carrots sold globally today are orange. So the domestication history of carrots suggests that the diversity of germplasm has already played a significant role in variation of nutrients that consumers get from the crop. I won't go through the details here, but there are white carrots which provide no nutrients but have significant fiber that are important for human health. Orange carrots with the vitamin A precursors, alpha and beta carotene. Sometimes they'll have purple pigments mixed with them. Red carrots with lycopene, the same pigments as in tomatoes, and yellow carrots with lutein, typical for other yellow vegetable crops that we eat. If we look at the history of carotenoid content, the content of these vitamin A compounds, the orange pigments of carrots in the U.S., there's been a significant increase in the content of these carotenoid pigments due to classical carrot breeding. In the 1970s, the average carrot was orange for sure, but only had about 90 parts per million of these pigments, whereas by 2010, the content of the average carrot sold in the U.S. based on statistics that have been kept over the decades, roughly 50% more than the orange carrots of the 1970s. So it's been possible through classical breeding to increase the content of these orange pigments. At the same time, we're interested in many other consumer quality factors that I mentioned before, like flavor and color and convenience. So this process of improving carrot colors by looking at the diversity of that variation that occurred in this wide range of carrots that I showed you in an earlier slide in this circle of different colored carrots. And of course, if you intercross different orange carrots, you're going to get orange carrots. And if you intercross in a breeding program, and if you intercross higher pigment orange carrots, you will be able to incorporate this trait. And this is our approach in improving carotene content in the last 40, 50 years for U.S. consumers. And this same trend has been occurring globally. So if we look at what the actual contribution of carrots to human health strictly on the basis of vitamin A, in 1975, roughly 7 to 8% of the vitamin A in the U.S. diet was estimated to come from carrots alone, which is quite a bit considering the average consumption is only less than 4 kilograms per capita per year. By 2005, and the numbers are similar today, about 12 to 13% of the vitamin A in the U.S. diet comes from carrots. The rest of it from other vegetables, fruits, and also from vitamin itself. It's been estimated that something like 60% of the vitamin A in European diets is coming from carrots, where the carrot consumption is it's higher than it is in the U.S. And so carrots are a potent source of nutrients, and this has been an important characteristic from our standpoint of carrot breeding, where we are utilizing the carrot genome then to improve the content of carrots. It's roughly 8 different genes that contribute to higher carotene content of carrots. We're tracking these with molecular markers in our breeding work. I mentioned flavor as an important trait as well, and contrary to this cartoon where the grocer says that all carrots are the same flavor, there's a wide range of carrot flavor. We pay close attention to this. As I mentioned, sugars play a critical part of carrot flavor, but also the typical flavor compounds we call carroty flavor vary quite widely as well. And we follow as we improve the crop. And there's roughly 15 to 20 genes that we're tracking with molecular markers in our breeding program today for the flavor traits in carrot improvement projects. So if we look at where we're at on improving consumer quality of carrots, there's a wide variation of traits for consumer traits. We have a better understanding of the genes controlling the carotenoid pathway in particular because of its importance for human health. But we also have put a lot of effort into tracking the genes for purple pigments, which I didn't talk about much, but purple pigments are terrific antioxidants and important for consumers for that reason. With the genome the improvement of flavor is probably most important. It's been estimated that less than a third of U.S. consumers eat their recommended servings of vegetables every day, and flavor is a major variable that limits the interest of consumers in vegetables. So we're paying a lot of attention to it in carrot improvement and tracking genes involved in the content and composition of the flavor profile of carrots. And these genomic tools to improvements, sugars and flavor are critically important as we're improving the crop, primarily from a U.S. consumer standpoint, but also with significant applications globally, especially in areas of the world where there's vitamin A deficiency. So in the last 50 years, there's been about a 6-fold increase in the production of carrots globally, while the global population has roughly doubled. The question then for me as a carrot breeder is what variation is out there that we can tap into to sustain the production of globally. In other words, what does the carrot grower need to keep growing the crop? And might there be new challenges that are showing up for carrot production in the future that growers aren't facing today? If we look at one variation on carrot breeding that's been significant that I'll only mention in this one slide, it's the development of carrot hybrids. I won't go into the details of that, but from a breeding standpoint, this has been critical, and most of the new cultivars of carrots that are released in most of the world today are hybrids. And this is also due to, attributable to the genetic variation of the crop, where in the 1950s, the genetic basis for the ability to make carrot hybrids was discovered. I'll not go into it anymore, but it was wild carrots actually that this trait for the development of hybrids changes the morphology of flowers that has been the basis for the development of hybrid carrots. So going back to what traits we're looking at as we are developing carrots to make hybrids, from a grower standpoint, growers face, like other crops, a wide range of challenges from pests and diseases. And so some major diseases are leaf blight and root knot nematodes, cavity spot. We've got some significant work underway evaluating major genes controlling alternary leaf and especially root knot nematodes, where there's we were concerned whether we could find nematode-resistant carrots that are resistant enough to withstand nematode attack. And in fact, there's strong genetic resistance for nematodes in the wide range of germplasm, which we're tapping into to improving that trait today. There are certainly other diseases, but in addition to diseases and pests, carrot growers want a crop that they can count on growing in a way without flowering, because the initiation of flowering ruins the consumer quality of carrots. And so this is referred to as bolting, and there is a significant genetic evaluation of genes for bolting or floral induction of carrots underway. Early seedling vigor is important for carrots. Carrots are notoriously slow growers early on, but there's genetic variation for this trait as well that we're evaluating with genetic markers in our breeding programs today. And also, as growers grow the crop, there's certainly an interest in lower input. Lower input agriculture is important for the economy of the growers, but also for the health of the planet. And so we're looking at seeing some significant variation in the need for nitrogen fertilizers and other nutrients in the soil necessary to grow carrots. Finally, we're looking at abiotic stress. This is something relatively new for carrots, not necessarily that new, but historically there's not been much attention paid to abiotic stress in carrots. When we look at this question of feeding global populations in the future, abiotic stress looks to be a bigger factor than what we've been paying attention to in the past when it comes to carrot improvements. And if we look at carrots as a crop, it's typically considered to be a cool season crop. And if we're looking at warmer, drier climates with more saline soils, what challenges can the grower be facing? Is there enough genetic variation there to improve the crop to keep up with the potential of global warming as we go along, or will carrot as a crop die off because of the challenges of growing it? So if we look at carrots as a crop, it's, as I said, it's typically characterized as a cool season crop with optimal temperatures of 7 to 24 centigrade, not very warm temperature at all. But let's take a look at where wild carrot comes from hot, dry regions of the world. So this is good news from a breeding standpoint, because when we look at carrots that come from places like the landscape of Uzbekistan or Tunisia, which are in these photos on the left where I selected wild carrots, these carrots, it turned out, have some greater tolerance to heat and drought than some of the other carrots that we've looked at in diverse carrot collections. This is important because if we look at the major carrot production area of the U.S. in Central California, typically there's been about 10 days a year where the temperature exceeds 80 centigrade, but in 2018, 60 days, 60, exceeded 38 C. So warm temperatures are coming to carrot growers in the U.S., and this is of great importance for sustaining crop production for the U.S. and globally. So why would a cool season crop be fairly well adapted to warm climates? It's because of its origins in Central Asia, where it's warm and dry. And so the domestication history of carrots is important and suggests that this diversity may be there. What's the evidence for this? There historically has been little evidence about heat, drought, or salinity tolerant carrots. But when we recently had a project in Pakistan and Bangladesh, my collaborators looking at a diverse collection of carrots. This is a carrot field you're looking at. It's not an empty field, but it's a carrot field with significant heat stress in Bangladesh. Only a few carrots grew. We were evaluating a fairly small collection, but about 10–15% of the entries demonstrated some heat tolerance, both wild and cultivated. Likewise, for drought tolerance and for salt tolerance. So the bottom line is there is the variation there that we can tap into to improve carrots, and now we're in the process of harnessing that variation so that growers in the future can rely on tapping into that variation that the seed companies and wheat breeders of carrots are working on to improve the crop for these traits. So in summary then, looking more broadly at the range of traits that are important for growers, as we had observed for consumer traits, there's broad genetic variation for most of those production traits. We're surprised to find the variation in early seedling growth because of the nature of just the general way that crops in the apiaceous crops group of plants grow. But in fact, we've found variation for early stand establishment and seedling growth. That'll be an important factor for carrot production in the future, in addition to resistance to disease that are commonly found in carrots globally. And also, like other crops, there are new pests and diseases that are showing up, insects, and some significant microbial diseases showing up in carrots that we're starting to look at variation for resistance to because of the appearance of these diseases in more high frequency globally than what we've seen in the past. As I mentioned before, the growers are interested also in having carrots that not only grow quickly early, but also we're looking at carrots that establish a fairly large top size, not too large, but enough to suppress weeds better than what typical carrots will grow. And I mentioned before the need for early bolting tolerance is controlled by a couple of genes, and we have selection underway in our breeding programs as we're improving the crop. And then of particular interest, the importance of improving abiotic stress tolerance will, I expect, play a particularly important role in carrot improvement in the future. And in this case, we're also finding a range of variation. Something like salinity carrot was historically thought to be very saline sensitive, but in fact, there's a wide range of salinity tolerance in carrots. And it's not simply inherited, but with the carrot genome, we now have the tools, and we've started developing the markers to select for abiotic stress tolerance in carrots. And with this genomics assisted breeding process, we in the public sector are developing these tools and working with the industry carrot researchers and breeders to better develop carrots for carrot growers and for carrot consumers in the future. So if we look at where we have come from with carrots, historically, as I mentioned earlier, some brave souls in Central Asia were bold enough to start eating wild carrots and cultivating them in that part of the world apparently first. And carrots grew as a crop globally in the last 1,100 years to the east and west of Central Asia. And there's a wide range of diversity in that crop. Wild carrots are fully crossable with cultivated carrots, and this is an important source of variation we will use for future improvements of carrots of all traits, but especially those abiotic stress traits. And this will be important because of the need for certainly larger populations in the future, as well as more climate challenges and no doubt diseases and other challenges showing up for growers in the future. Where the sequencing of the genome has been already important in discovering of traits and genes for traits that are important for carrot improvement that we're implying in breeding programs today. There's a need for improvement in other apiaceous crops that I mentioned, and you're well familiar with as well, crops like celery, coriander, fennel, dill, and the information we're gathering from carrots to some extent will be applicable to these crops as well. And we so far have put a big focus on biosynthesis, in particular the carotenoids, but also the anthocyanins, because of not only their importance to the appearance of carrots to consumers, but in fact the quality of carrots to consumers as a source of vitamin A in the case of the orange pigments, but other nutrients in yellow, red, and white and purple carrots as well. So with that, I think I've come to the end of my slides for my presentation, and as there may be questions, I would be glad to field those. Thank you for your time and your interest in hearing this talk.
Thank you very much, Professor Simon. Thank you for your valuable and informative presentation. Is there any question? I think no. Okay. Yes, I think this session is finished. I hope the next meeting will be face-to-face. I would like to thank distinguished invited speakers for attending the TÜRKİYE 7th Seed Congress. Many thanks for the participants who have waited until this time. And many thanks also Dr. Eren and committee members. I will give the word to Dr. Eren. Yes, Eren. Dr. Eren, please go ahead.
Thank you, Professor. I gave a short speech at the beginning of the session, but since some of the speakers have just joined, I would like to speak again. Good evening to us from Turkey, and good afternoon, Dr. Nagel from Germany and Dr. Lögner from United Kingdom. And good morning, Dr. Tyler, Dr. Simon, and Dr. Welbaum from United States. I hope you are all having a good day so far. My name is Eren Özden. I am the chair of the Congress organizing team. I am very excited. Thank you very much for accepting our invitation and sharing your valuable knowledge with us. We had a very exciting afternoon with a very special guest. We are very happy and honored you were able to join us. Thank you very much for your time and attention, Dr. Nagel, Dr. Tyler, Dr. Löbner, Dr. Futit, Dr. Welbaum, and Dr. Simon. Thank you.
Okay, have a nice evening and have a nice day.